There exists, in the quiet corners of the natural world, an order of creatures so ancient, so architecturally sophisticated, and so profoundly misunderstood that even the most seasoned naturalists pause in genuine reverence before them. Spiders, members of the order Arachnids, represent one of the oldest and most enduringly successful lineages of predatory arthropods to have ever colonized the terrestrial realm. Their fossil record extends back more than 380 million years, predating the emergence of dinosaurs by nearly 150 million years, and yet their fundamental body plan has remained remarkably stable across geological epochs. This conservatism in form speaks not to evolutionary stagnation, but rather to an extraordinary degree of adaptive perfection. The two primary anatomical regions of a spider, the prosoma, which houses the brain, eyes, and limbs, and the opistosoma, which contains the organs of digestion, reproduction, and silk production, together constitute a biological architecture of breathtaking economy and efficiency, one that nature has seen little reason to fundamentally revise across hundreds of millions of years of continuous existence. The silk-producing apparatus of spiders is, without exaggeration, one of the most remarkable biological innovations in the history of multicellular life on Earth. Located at the posterior of the opistosoma are specialized glandular structures known as spinnerets, typically 3 pairs in most species, though some primitive lineages retain 4, which are connected internally to a complex array of different silk-producing glands. Each of these glands synthesizes a chemically distinct form of silk protein, called spidroin, tailored to specific functional requirements. The ampullate glands produce the structural dragline silk used for locomotion and the radial threads of orb webs, while the tubuliform glands produce the coarser silk used to encase egg sacs. The aciniform glands generate wrapping silk for immobilizing prey, and the aggregate glands, present only in certain lineages, produce the sticky, adhesive globules that transform spiral capture threads into effective traps. The architectural coordination required to deploy these different silks simultaneously and with precision during web construction represents a feat of neurological and mechanical sophistication that continues to astonish biologists and materials scientists alike. The chemistry of spider silk is, from a molecular perspective, a masterwork of protein engineering achieved through hundreds of millions of years of evolutionary refinement rather than deliberate design. The primary structural proteins, the spidroins, are notable for their extreme molecular weight, often exceeding 1 million daltons, and for their unusual amino acid composition, which is dominated by glycine and alanine residues arranged in highly repetitive motifs. These repetitive blocks adopt 2 distinct secondary structures depending on their precise sequence: beta sheet crystalline regions, which provide tensile strength and resist stretching, and glycine-rich helical regions, which confer elasticity and allow the fiber to absorb energy without catastrophic fracture. The result is a material that achieves a combination of strength and toughness that no synthetic fiber produced by human industry has yet managed to replicate simultaneously. Dragline silk from the golden silk orb-weaver, Nepheloclavipes, has been measured at a tensile strength approaching 1.3 gigapascals, comparable to high-grade steel on a weight-for-weight basis, while possessing an extensibility and energy absorption capacity that steel cannot approach. Among the approximately 48,000 described species of spiders that currently inhabit our planet, an extraordinary diversity of web architectures has evolved, each representing a distinct solution to the universal challenge of capturing prey with minimum energetic expenditure. The orb web, that familiar geometric masterpiece of concentric spirals supported by radiating structural threads, is perhaps the most recognizable, yet it represents only one of at least a dozen fundamentally distinct web types that arachnologists have identified and formally classified. Sheet webs, funnel webs, cobwebs, tubular webs, tangle webs, and domed webs each solve the problem of prey interception through radically different geometries and material strategies. Some sheet web spiders construct horizontal platforms with a chaotic tangle of trip wires above them, causing flying insects to lose altitude and tumble onto the adhesive surface below. Others build downward opening funnels that draw prey inward toward a central retreat tube where the spider waits in concealed readiness. Each architectural strategy reflects a deep evolutionary conversation between the spider's sensory capabilities, behavioral repertoire, prey availability, and the physical constraints of its particular microhabitat. The evolution of venom in spiders represents one of the most consequential biological innovations in the history of terrestrial ecosystems, fundamentally reshaping the dynamics of predator-prey interactions across virtually every land biome on Earth. Venom is estimated to have evolved independently in the arachnid lineage, leading to spiders at least once, early in their evolutionary history, and the chemical complexity of spider venoms has since diversified to an almost staggering degree through continuous arms race dynamics with evolving prey species. Spider venoms are complex mixtures of molecules including neurotoxic peptides, cytotoxic proteins, enzymes, polyamine compounds, and free amino acids, the precise composition varying enormously between species and even between individual spiders of the same species depending on age, diet, and geographic location. Of the nearly 48,000 described species, only a small fraction, perhaps 200 or so, possess venom that poses any meaningful medical risk to healthy adult humans. The majority of spiders are either too small to penetrate human skin effectively, possess venom too dilute or compositionally unsuited to cause significant systemic effects, or are simply so disinclined toward biting large vertebrates that envenomation under normal circumstances essentially never occurs. The black widow spiders of the genus Latrodectus occupy a singular position in the cultural imagination of the Western world, simultaneously demonized and romanticized, yet their biology is considerably more nuanced than popular mythology suggests. Distributed across temperate and tropical regions worldwide, with roughly 31 recognized species, Latrodectus spiders are characterized by their distinctive cobweb architectures, their sexual size dimorphism, females often 5 times the mass of males, and of course by the potent neurotoxic venom that has made them famous. The primary toxic component of their venom, alpha-latrotoxin, is a large protein molecule that binds irreversibly to presynaptic receptors at neuromuscular junctions, triggering a massive and sustained release of neurotransmitters that causes the syndrome known as latrodectism. Symptoms include intense localized pain spreading to regional lymph nodes, muscle rigidity, sweating, nausea, and hypertension, a constellation that is extremely unpleasant but, in otherwise healthy individuals receiving appropriate medical attention, very rarely fatal. The famous sexual cannibalism of Latrodectus females, wherein males are sometimes consumed following mating, occurs far less commonly in nature than popular accounts imply, with many males successfully escaping after copulation. The Brazilian wandering spiders of the genus Phenutria, whose name derives from the Greek for murderous, are broadly considered among the most medically significant spiders in the world, not merely for the potency of their venom but for their behavioral ecology, which brings them into frequent and sometimes aggressive contact with human populations. Unlike most spiders that flee from confrontation, Phenutria species adopt a distinctive threat display when disturbed, raising their front legs high and swaying from side to side before striking, a behavior that has earned them the colloquial name armed spiders in Brazil, where they are most diverse. Their venom contains a particularly complex mixture of neurotoxic peptides, including the PHTX toxins, that act on both sodium and calcium ion channels, producing a syndrome that may include intense localized pain, systemic effects including tachycardia and hypertension, and in rare cases, particularly in children or immunocompromised individuals, potentially dangerous respiratory compromise. A remarkable and medically intriguing property of certain Phenutria venom components is their capacity to induce priapism, a prolonged and painful penile erection, a side effect that has attracted significant pharmaceutical research interest in the development of treatments for erectile dysfunction. The Sydney funnel web spider, Atrax robustus, commands a unique position in the hierarchy of medically dangerous spider species, distinguished not merely by the lethality of its venom but by a set of biological peculiarities that make it exceptionally hazardous to humans, specifically among all primates. Found in a relatively restricted range in and around the Greater Sydney Basin of southeastern Australia, Atrax robustus constructs distinctive funnel-shaped webs in moist, sheltered locations, beneath logs, in rock crevices, and frequently in suburban gardens. The venom of adult male Atrax robustus contains atricotoxins, particularly delta-atricotoxin, which causes massive activation of voltage-gated sodium channels, overwhelming the autonomic and somatic nervous systems simultaneously. A striking and evolutionarily puzzling feature of this venom is its remarkable potency in primates, including humans, compared to its relatively modest effects on many non-primate mammals. This selectivity appears to relate to structural differences in primate sodium channel subtypes that make them disproportionately sensitive to atricotoxin binding. Fortunately, the development of an effective antivenom in the early 1980s has essentially eliminated fatalities from Atrax robustus bites in Australia, transforming what was once a genuinely life-threatening encounter into a manageable medical event. Entirely distinct from the neurotoxic venoms of spiders like Latrodectus and Atrax are the cytotoxic venoms of the recluse spiders, genus Loxosceles, which attack tissues directly rather than targeting the nervous system. The brown recluse, Loxosceles reclusa, most notorious among the approximately 100 species in the genus, is native to the south-central United States and is characterized by its distinctive violin-shaped marking on the cephalothorax, its reduction to 6 eyes arranged in 3 pairs rather than the typical 8 of most spiders, and its preference for dry, undisturbed environments such as closets, attics, and spaces beneath wooden furniture. The primary agent of tissue damage in Loxosceles venom is sphingomyelinase D, an enzyme that cleaves sphingomyelin in cellular membranes, triggering a cascade of inflammatory responses and complement activation that can result in a necrotic skin lesion, the condition known as loxoscelism. The severity of this reaction varies enormously between individuals and bite events, with many bites causing minimal local tissue damage that heals without medical intervention, while a smaller proportion develops significant necrotic wounds. Systemic loxoscelism, involving kidney damage and hemolysis, is rare but represents a genuine medical emergency requiring urgent hospitalization. The tarantulas, family Theraphosidae, represent what many arachnologists consider the most architecturally primordial of the large spider lineages that persist in significant diversity today, having changed remarkably little in fundamental form from ancestors that walked the Carboniferous forests more than 300 million years ago. With over 1,000 described species ranging across tropical and subtropical regions of every continent except Antarctica, Theraphosids exhibit an extraordinary ecological diversity belied by their superficially similar appearance. They range in size from species with body lengths of barely 2 centimeters to the Goliath bird eater, Theraphosa blondi, of South America, whose leg span may exceed 28 centimeters and whose body mass can reach 170 grams, making it the heaviest spider known to science. Despite their formidable appearance and reputation, most tarantulas are remarkably reluctant to bite, preferring instead to flee from disturbance or, in the case of New World species, to deploy their most distinctive defensive adaptation, the urticating hairs that cover the opisthosoma and which can be rapidly and forcefully kicked loose using the rear legs, creating a cloud of microscopic barbed fibers capable of causing intense irritation to the eyes and mucous membranes of potential predators. The evolution of urticating hairs, or more precisely, urticating setae, in New World tarantulas represents one of the most elegant examples of a defensive innovation arising through the modification of pre-existing structures in the entire history of arachnology. These specialized hairs, found only in the approximately 800 species of theraphosid tarantulas native to the Americas, are derived from the same sensory and integumentary structures present across all spiders, but have been modified through evolutionary time into weapons of extraordinary effectiveness. Ranging across 8 recognized morphological types designated by Roman numerals, urticating setae differ in their barb geometry, penetration capacity, and the specific tissues they most effectively irritate. Type 1 hairs, present on species like Brachypelma hamorii, the Mexican red-knee tarantula, are effective primarily against mammalian facial tissues. Type 3 setae, found in Theraphosa blondi and certain other large South American species, possess backward-curving barbs capable of penetrating deeper into ocular tissues and causing severe ophthalmological complications. The behavioral deployment of these setae, a rapid, deliberate stroking of the opisthosoma with one or more rear legs to dislodge a directed cloud of fibers, represents a behavioral module distinct from the prey capture and web construction behaviors that characterize most spider defensive repertoires. Among the most remarkable of all spider behaviors is the phenomenon of ballooning, a mode of aerial dispersal in which spiders, typically small juveniles or adult dwarf species, release silk threads into the atmosphere and are carried by air currents over potentially vast distances. The physics and precise mechanism of ballooning were subjects of scientific debate for centuries, with early observers noting that the silk threads deployed during ballooning events appeared to be influenced by electrostatic forces as well as wind, an observation that was long considered speculative but has more recently received rigorous experimental validation. Research published in the early 21st century demonstrated definitively that atmospheric electric fields, the same fields responsible for driving charge from the ionosphere toward the Earth's surface, can exert sufficient upward force on the negatively charged silk filaments of ballooning spiders to lift the spiders against mild downdrafts, explaining observations of spiders becoming airborne in completely calm conditions. Spiders have been collected alive at altitudes exceeding 4,000 meters and found on oceanic research vessels hundreds of kilometers from the nearest land, demonstrating the extraordinary range over which ballooning serves as a dispersal mechanism. This capacity for long-distance aerial colonization helps explain the remarkably cosmopolitan distributions of many spider lineages. The sensory world of a spider is profoundly alien to human intuition, constituted primarily not through vision, though some groups have evolved remarkably acute visual systems, but through an extraordinarily sensitive suite of mechanoreceptive organs distributed across the body surface and appendages. The slit sensilla, microscopic stress-sensitive slits in the exoskeleton connected to sensory neurons, are present on virtually every appendage and body region in spiders and function collectively as an organ of exquisite sensitivity to substrate vibrations, air currents, and mechanical deformations. Trichobothria, the fine hair-like sensilla projecting from the leg surfaces, can detect air particle oscillations caused by nearby flying insects with extraordinary sensitivity, with some species capable of detecting motion at frequencies corresponding to the wing beats of small dipterans at distances of several centimeters. Collectively, these vibration-sensing systems allow a spider sitting at the hub of its web to extract a remarkable amount of information about objects making contact with the web, including the size, location, and even the species identity of prey, from the pattern of vibrations transmitted through the silk threads. Web-borne vibration thus functions as a kind of extended sensory organ, an externalized nervous system of extraordinary sensitivity and informativeness. The eyes of spiders present one of the most fascinating case studies in the evolution of visual systems across the animal kingdom, exhibiting an enormous range of complexity, optical design, and functional sophistication that mirrors the extraordinary ecological diversity of the order. Most spiders possess 8 eyes arranged in 2 rows, though the precise configuration, relative size, and optical design of these eyes varies considerably between major taxonomic groups and reflects the very different ecological roles visual information plays in their respective lifestyles. Web-building spiders that rely primarily on vibrational information from their webs typically possess eyes of quite modest acuity, used principally for detecting the gross movements of large potential predators rather than for fine-grained visual analysis of the environment. Jumping spiders of the family Salticidae, by dramatic contrast, have evolved a visual system of extraordinary sophistication that represents one of the most remarkable optical architectures in the animal kingdom. Their large, forward-facing principal eyes, designated the anterior median eyes, are telephoto systems with a narrow field of view but exceptional resolving power, achieving spatial acuity that approaches or matches that of much larger vertebrates, despite fitting into a head only a few millimeters across. This achievement is enabled by a layered retina with multiple photoreceptor types and a uniquely mobile retinal tube that scans the visual field. The jumping spiders, family Salticidae, with over 6,500 described species, the largest family of spiders on Earth, have earned a distinguished reputation among arachnologists and behavioral ecologists for the complexity and apparent intentionality of their behavior, which has been the subject of increasing scientific scrutiny over the past several decades. Unlike most spiders, which respond to prey primarily through vibrational cues and reflex-mediated capture behaviors, salticids engage in elaborate visual stalking behaviors that involve detour navigation, perspective taking, and evidence of what appears to be forward planning, cognitive capacities that were long considered exclusive to vertebrates. In a landmark series of experiments conducted by Fiona Cross and Robert Jackson at the University of Canterbury, the salticid Portia fimbriata was shown capable of navigating complex detour routes to reach prey when the most direct path was obscured, a task that appeared to require the spider to maintain a mental representation of the prey's location throughout the detour. Portia fimbriata also demonstrates sophisticated predatory versatility, specializing in the challenging task of hunting other spiders within their own webs, a feat that requires both exceptional vibratory mimicry skills to create signals within the web that lure the resident spider toward the predator, and the behavioral flexibility to modify these signals adaptively based on the resident's responses. The sexual biology of spiders is among the most complex, varied, and evolutionarily contentious of any arthropod order, encompassing phenomena ranging from elaborate multimodal courtship displays to traumatic insemination, limb autotomy, and the contested phenomenon of sexual cannibalism. Male spiders face a fundamental challenge imposed by their anatomy. Unlike most other animals, they lack intromittent organs on the genital segment and instead transfer sperm indirectly, first depositing a sperm web from which they charge the paired palpal organs, the pedipalps, with sperm, then inserting these modified appendages into the female's paired genital openings during mating. The pedipalp is an extraordinarily complex structure, particularly in more derived spider families, where it may contain hydraulically operated sclerotized components, coiled embolus tubes, and various secondary structures that appear to function as mechanical keys specific to the complementary structures of the female's epigenum, the genital plate, potentially serving as species-isolating mechanisms that prevent hybridization between closely related species. In some groups, notably the orb-weavers of the family Arachnidae, males break off one or both palps within the female's genital ducts during mating, leaving them in place as copulatory plugs that impede subsequent insemination by rival males, at the cost of the male's future reproductive capacity but potentially increasing his current mating success. Sexual cannibalism in spiders, the consumption of males by females before, during, or after copulation, is among the most widely recognized yet poorly understood phenomena in behavioral ecology, surrounded by a rich literature of conflicting hypotheses regarding its adaptive significance and evolutionary maintenance. While dramatically portrayed in popular accounts and even in some scientific literature as a near-universal feature of spider mating, systematic observational studies reveal that sexual cannibalism rates in nature are far more variable than commonly implied, ranging from essentially absent in some species to approaching 80% of mating attempts in others under natural conditions. In species like the Australian redback spider, Latrodectus hasselti, males actively somersault into the female's chelicerae during copulation, a behavior that has been interpreted as adaptive mate sacrifice, with cannibalized males gaining prolonged copulation duration, increased sperm transfer, and potentially enhanced offspring provisioning through the nutritional contribution of their body mass to the gravid female. The evolutionary dynamics of sexual cannibalism are complicated by the fact that female aggressiveness, male escape behavior, and environmental context including prey availability, male body condition, and operational sex ratio all interact in determining cannibalism rates, making simple adaptive narratives difficult to sustain across the full diversity of spider mating systems. Among the most astonishing reproductive strategies in the entire spider order is the phenomenon of matrophagy, the deliberate consumption of the mother by her offspring, documented in several taxonomically distinct lineages and representing one of the most extreme forms of parental investment described in any animal taxon. In the colonial spider Stegotyphus demicola of southern Africa, and in the independently evolved matrophagy systems of several other Stegotyphus species as well as the unrelated genus Diea in Australia, the mother undergoes a systematic physiological transformation following the emergence of her offspring from the egg sac. Her digestive organs liquefy as she regurgitates increasing quantities of nutrient-rich fluid for her spiderlings to consume, simultaneously allowing them access to her body tissues. This process of progressive liquefaction and consumption, which the mother survives for days or even weeks, represents the ultimate form of terminal investment in offspring quality, converting the entire biological substance of the maternal body into a concentrated nutritional resource for the next generation at the precise moment when that generation is most vulnerable and most in need of high-quality nutrition to fuel their initial growth and development. The spiderlings produced in matriphagy-practicing populations tend to be significantly larger and have higher survival rates than those in populations where matriphagy has been experimentally prevented. The social spiders, a loose grouping of species from several unrelated families that have independently evolved various degrees of cooperative living, represent some of the most scientifically compelling and biologically counterintuitive members of the entire order, given that the overwhelming majority of spiders are profoundly asocial and often aggressively cannibalistic toward conspecifics. True sociality in spiders, defined by the permanent tolerance of multiple adults including adult females within a shared web structure, the cooperative capture of prey, cooperative brood care, and overlapping generations, has evolved independently at least 23 times across at least 18 different genera. A striking degree of parallel evolution that suggests powerful selective pressures favoring cooperative living under specific ecological conditions. These conditions appear to include habitats with high prey availability but individually unmanageable prey sizes, environments where web construction is particularly expensive due to limited suitable sites, and ecological contexts where predation pressure on solitary individuals is intense. The colonial Anolosomus eximius of the South American tropics can form supercolonies containing tens of thousands of individuals across web structures spanning many cubic meters, with individual colony members showing behavioral specialization, though not morphological differentiation, related to prey capture, web maintenance, and brood care, suggesting incipient task allocation without the morphological caste system characteristic of insect eusociality. The question of how social spiders maintain genetic cohesion within their often highly inbred colonies has generated substantial scientific discussion, as most social spider species reproduce predominantly through sibmating within the natal colony, with dispersal events being relatively rare and typically involving small groups of related individuals. This extreme inbreeding, which would be catastrophic in most animal populations, appears to be tolerated, or even adaptive, in social spiders through mechanisms that remain incompletely understood but likely involve purging of deleterious recessive alleles from the population, potential local adaptation of inbred lineages to specific ecological conditions, and the benefits of high genetic relatedness in promoting altruistic cooperation among colony members. Population genetic analyses of social spider colonies typically reveal fixation index values approaching 1, indicating near-complete homozygosity, yet colonies do not consistently show the fitness reductions predicted by standard inbreeding depression models. Research on Stegotyphus mimisarum in Africa has suggested that colony-level selection may operate on these highly related groups in ways that resemble individual-level selection in more outbred populations, effectively treating the colony as a superorganism whose inclusive fitness is maximized through the coordinated behaviors of its highly related members. A theoretical framework that has profound implications for our understanding of the conditions necessary for the evolution of cooperative behavior. The bolus spiders of the genus Mestophora constitute one of the most breathtaking examples of chemical mimicry and specialized predatory deception in the entire animal kingdom, representing a level of evolutionary sophistication that continues to inspire wonder among entomologists, behavioral ecologists, and evolutionary biologists working across multiple disciplines. Rather than constructing conventional webs, female Mestophora spiders hunt nocturnally by dangling a single thread of silk terminating in a sticky globule, the bolus, and swinging this device to intercept approaching male moths. The extraordinary specialization of this system lies in the chemical composition of the sticky globule, which contains synthesized analogues of the female sex pheromones produced by specific moth species. These pheromone mimics lure male moths through olfactory deception and, remarkably, different Mastophora species target different moth species, and individual spiders have been observed to shift their pheromone composition across the course of a single night, sequentially attracting males of different moth species that are active at different periods during darkness. This suggests that the spiders possess some capacity to produce multiple pheromone compounds and regulate their emission temporally, a degree of chemical sophistication that requires not only the biosynthetic machinery to produce multiple complex volatile molecules but also the behavioral control to deploy them adaptively across changing ecological contexts. The trapdoor spiders, a polyphyletic grouping distributed across several families including Tenozidae, Idiopodidae, and Actinopodidae, construct some of the most architecturally sophisticated burrow systems in the arthropod world, combining precise engineering, patient ambush strategy, and remarkable longevity in a predatory lifestyle that has proven extraordinarily successful across tropical and warm-temperate regions of every inhabited continent. These spiders excavate burrows in soil using their chelicerae and rake-like spines on the legs, lining the tunnel walls with silk to stabilize and waterproof the structure, then constructing a hinged door of soil, silk, and plant material that fits its frame with astonishing precision and is weighted or held shut against intruders. The spider waits just below the closed door, holding it shut with its forelegs while simultaneously monitoring for prey vibrations through the substrate and through silk trip lines radiating from the door. When suitable prey is detected passing nearby, the door is thrown open and the prey seized in a movement measurable in milliseconds, one of the fastest prey capture events in the spider world. A particularly remarkable feature of trapdoor spider biology is their exceptional longevity. The recently documented death of a wild trapdoor spider designated number 16 from a long-term study site in Australia at an estimated age of 43 years made headlines worldwide and established a new record for spider longevity. The diving bell spider, Argyroneta aquatica, stands alone among the approximately 48,000 known spider species as the only member of the order to have successfully colonized a fully aquatic lifestyle, spending the overwhelming majority of its adult life submerged in freshwater habitats including ponds, slow streams, and fin pools across Europe and Central Asia. Its survival beneath the water surface depends on a remarkable application of silk technology. The spider constructs a dome-shaped retreat of silk anchored to aquatic vegetation, then makes repeated surface trips to collect air bubbles trapped among the abdominal hydrophobic hairs, which it carries down and releases inside the dome. The cumulative trapped air creates a self-contained atmospheric pocket that the spider inhabits like a miniature terrestrial environment, hunting, eating, molting, mating, and laying eggs entirely within this silken submarine home. The diving bell functions not merely as a contained air reserve but as a genuine physical gill. Oxygen diffuses into the trapped air bubble from the surrounding water down its concentration gradient, while carbon dioxide diffuses outward, extending the functional life of the trapped gas far beyond what would be predicted from a simple sealed air model. Research has demonstrated that a well-constructed Argyroneta diving bell in well-oxygenated water can extract sufficient dissolved oxygen from the surrounding medium to support the spider's metabolic needs essentially indefinitely without requiring surface replenishment. The mirror spiders of the genus Thwytesia and the related genus Gastracantha in different contexts possess one of the most visually arresting appearances of any spider on Earth, their abdomens bearing patches of reflective, silver-colored guanine crystal deposits beneath a transparent cuticle that create an appearance of scattered fragments of broken mirror embedded in the living tissue of the opisthossoma. This striking visual quality, which has made Thwytesia species among the most photographed of all small spiders in the age of macro photography and social media naturalism, serves a biological function that researchers have only recently begun to investigate with scientific rigor. The guanine crystals, a metabolic waste product of purine catabolism that many spiders store rather than excrete, are present in many species but in Thwytesia have been elaborated into unusually large, flat, highly reflective structures. The adaptive significance of this reflectivity appears to involve disruptive coloration that breaks up the spider's outline when viewed against complex natural backgrounds such as lichen-covered bark, and possibly involves mimicry of light reflections from dewdrops or water surfaces. The silver patches are not static. They can be actively contracted or expanded by muscular action, reducing their area by up to 80% when the spider is alarmed, a dynamic color change that suggests a complex signaling role in addition to any passive camouflage function. The genus Dolomites, the fishing spiders of the family Pisoridae, has evolved one of the most remarkable ecological extensions of the spider body plan, exploiting the surface tension of still freshwater bodies as both a locomotory platform and a hunting ground in ways that reveal the extraordinary physical sophistication possible within the constraints of an arachnid body. These large, semi-aquatic spiders rest with their legs spread on the water surface, exploiting the surface film to support their mass while simultaneously using this film as a highly sensitive vibrational detection system that allows them to locate aquatic prey moving beneath the surface. When a disturbance is detected, from an injured or struggling insect, a small fish, or a tadpole, the spider can pursue prey across the water surface at remarkable speed, and can even plunge through the surface film to seize aquatic prey up to several times its own body mass from below. The physical feat of moving across water while simultaneously hunting through it requires exquisite coordination of limb movements to manage the dynamics of surface tension interaction, and Dolomites spiders achieve this through leg movement patterns that are measurably different from those used on solid substrates. Remarkably, Dolomites can also exploit water surface waves for communication during courtship, creating and detecting specific wave patterns that carry information about the signaler's size, sex, and reproductive status. The net-casting spiders of the family Deinopodidae, colloquially known as ogre-faced spiders on account of their enormous forward-facing principal eyes, have evolved what is arguably the most specialized active prey capture technique in the entire spider order, constructing a miniature rectangular web of extraordinarily elastic hackled band silk between their front forelegs and lunging this net over passing prey with a speed and precision that represents one of the most spectacular predatory behaviors in the invertebrate world. The eyes that give these spiders their common name are the anterior median eyes, principal eyes grown to truly extraordinary proportions relative to body size, with lenses so large and light-gathering that they provide exceptional visual sensitivity in near darkness, enabling the spider to detect and track prey in conditions approaching absolute visual threshold for vertebrate eyes. The light-gathering membranes of these enormous eyes are constructed afresh each night from thousands of precisely arranged photoreceptor membrane tubes, and then destroyed and reabsorbed by the spider each morning before dawn, an extraordinary daily cycle of biological construction and demolition driven presumably by the vulnerability of such large, elaborate photoreceptor membranes to ultraviolet photodamage during daylight. The spitting spiders of the family Cytididae have evolved a prey capture strategy so entirely unlike that of any other spider family that they deserve consideration as one of the most remarkable evolutionary experiments in the history of the order. Rather than capturing prey through web architecture, chemical lures, or pursuit, Cytode species project a stream of venomous, adhesive silk from their chelicerae at prey from a distance of up to 2 centimeters, pinning the target to the substrate before it can escape or retaliate, a capability that extends their effective predatory range beyond the contact distance typical of most spiders and allows them to immobilize prey that might otherwise escape or inflict defensive injury. The silk and venom mixture is produced in massively enlarged glands of the cephalothorax. The characteristic domed shape of Cytode spiders reflects these enlarged glands, and is ejected by rapid muscular compression of the gland while the chelicerae vibrate rapidly, creating the characteristic side-to-side zigzag pattern of the projected stream. The entire projection event is completed in approximately 1.1 milliseconds, far too fast for the prey to detect and respond to before impact. What makes this strategy particularly elegant is that the adhesive quality of the silk increases dramatically upon contact with the substrate, creating a three-dimensional lattice structure that physically constrains the target through both mechanical entanglement and chemical adhesion, allowing even large and potentially dangerous prey to be immobilized safely from a distance. The Goliath bird eater, Theraphosa blondi, has achieved a degree of popular fame and notoriety that far exceeds what one might expect from a creature whose ecological role in the rainforests of the Guiana highlands of South America is broadly similar to that of many other large terrestrial predators. As the world's heaviest spider, with exceptional individuals reaching body masses approaching 175 grams and leg spans exceeding 28 centimeters, Theraphosa blondi occupies an ecological position closer to that of a small mammalian predator than to the stereotypical image of a spider lurking in a modest web corner. Its primary prey consists of large invertebrates, earthworms, crickets, beetles, but the species does occasionally take small vertebrates including frogs, lizards, mice, and on very rare documented occasions, small snakes, earning it the dramatic common name that overstates its avian predation significantly. When threatened, Theraphosa blondi produces a distinctive threat sound through stridulation, rubbing of opposing body surfaces, audible to the human ear at distances of several meters, a behavior that appears to serve as an honest signal of the spider's size and defensive capability. Local indigenous communities in the Guiana highlands have long incorporated Theraphosa blondi into their nutritional landscape, cooking and consuming these spiders by singeing off their urticating hairs over flame and roasting the body in banana leaves, describing the flavor as prawn-like. The ancient mythology surrounding spiders constitutes one of the richest and most geographically widespread bodies of arachnid-related cultural material in human history, extending across virtually every inhabited continent and reflecting the prominent place of spiders in human perceptual experience across the full span of documented and undocumented cultural traditions. Perhaps the most extensively elaborated spider mythological tradition in the Western canon centers on the figure of Arachne from Greco-Roman mythology, a mortal weaver of extraordinary skill who, in the version most elaborately told by Ovid in his Metamorphoses, challenged the goddess Athena to a weaving contest, produced work of surpassing quality that the goddess could find no fault in, and was transformed into the first spider as punishment for her hubris in daring to compete with it. Divine. The Ovidian account is notable for the ambiguity it preserves regarding whether Arachne's transformation is punishment or mercy. Ovid describes Athena feeling pity as well as wrath, and for the detail that Arachne's tapestry, when it was complete, depicted the loves and transformations of the gods with unflattering realism, suggesting that the weaver's offense was as much one of truthful representation as of competitive audacity. The very word arachnida, the class encompassing all spiders, scorpions, mites, and their relatives, is derived directly from this mythological figure. Across the indigenous traditions of sub-Saharan Africa and their diaspora expressions throughout the Americas and Caribbean, the spider figure most richly elaborated in narrative tradition is Anansi, the trickster spider of Akan mythology originating among the Ashanti people of present-day Ghana, whose tales traveled across the Atlantic with enslaved African peoples and spread across the Caribbean and North American continent in a remarkable process of cultural persistence and creative adaptation. Anansi, whose name derives from the Twi word for spider, is a figure of extraordinary narrative complexity, at once cunning and foolish, generous and selfish, triumphant and humiliated, embodying the full spectrum of human moral possibility within a single trickster persona. In the original Akan tradition, Anansi is credited with bringing stories themselves into the world. He is not merely a character in folktales but the mythological origin of storytelling as a human practice, having purchased from the sky god Niam the ownership of all stories in exchange for capturing 4 notoriously uncatchable creatures through elaborate schemes of cunning and misdirection. The dispersal of Anansi stories through the African diaspora represents one of the most striking examples of cultural survivorship through the Middle Passage, with Anansi tales documented in Jamaica, Trinidad, Suriname, Curaçao, and the coastal southeastern United States, each regional tradition bearing both clear continuity with West African originals and creative local adaptations reflecting new geographic and social contexts. The spider mythology of the indigenous peoples of the North American Great Plains and Southwest constitutes a profoundly different cultural engagement with arachnid symbolism from either the Greco-Roman or West African traditions, with spider grandmother, known as Kokiangwudi in Hopi tradition and by various names across other Pueblo and Plains cultures, occupying a role not of trickster or tragic heroine but of cosmogonic creator and spiritual guide. In Hopi cosmology, Spider Grandmother is among the first beings to exist and participates directly in the creation of humanity, In some accounts, she fashions the first humans from clay and breathes life into them through song, and in others, she guides the first people upward through successive underworld realms to reach the present world. The spider's web in these traditions functions as a cosmological metaphor of extraordinary sophistication, representing the invisible connections that link all living beings within the fabric of creation, with Spider Grandmother as the weaver and sustainer of these fundamental relational bonds. This vision of the spider as creative force, maker rather than merely hunter, reflects a deep observational engagement with spider behavior that recognized the creative act of web building as metaphorically resonant with the construction of the world itself, an insight that appears to arise independently across multiple unconnected cultural traditions precisely because it responds to a genuinely striking quality of spider biology. The silk gland diversity within a single spider species is remarkable enough to challenge any simple conception of silk as a single substance, with the major ampullate, minor ampullate, tubuliform, aciniform, piriform, flagelliform, and aggregate glands each producing chemically distinct protein polymers whose properties are tailored through evolutionary refinement to their specific functional contexts. The flagelliform gland's silk deserves particular attention as one of the most mechanically extraordinary biological materials known, constituting the spiral capture threads of orb webs and possessing a combination of properties that places it in a category essentially unique among structural biomaterials. Unlike the stiffer dragline silk of the major ampullate gland, which resists extension but eventually fails brittly when stretched beyond approximately 30% of its original length, flagelliform silk can be extended to more than 200% of its resting length before failure while still recovering elastically to its original dimensions. This extraordinary extensibility is achieved through a molecular architecture in which the spidroin chains adopt a highly disordered, random coil configuration in the as-spun state, storing elastic energy by resisting entropic conformational changes upon extension and releasing this energy upon relaxation. The combination of this elastic capture spiral with the rigid structural frame of the web creates a functional architecture that maximizes energy absorption from prey impact, minimizing prey bounce back while maximizing retention. The biomechanics of orb web construction represent an engineering challenge of extraordinary complexity that has attracted sustained scientific interest from arachnologists, materials scientists, and engineers working at the interface of biology and applied physics. A typical orb web, produced by a spider in the range of Araneus diadematus, the European garden spider, contains more than 20 meters of silk arranged in a precise geometric pattern optimized for prey capture, prey retention, and structural integrity under wind loading, and is constructed in a remarkably consistent sequence of behavioral operations that transforms a point in space into a functioning two-dimensional filter for flying insects in approximately 30 to 60 minutes. The construction sequence begins with the establishment of the frame threads and radii through a complex series of bridge threads and primary frames built using the spider's own body weight and gravity as engineering tools, then proceeds through the construction of a temporary auxiliary spiral from the hub outward, and finally involves the removal of this temporary scaffold as the permanent sticky spiral is laid down from the periphery inward using it as a spacing guide. Computer modeling of web construction has revealed that the behavioral program controlling this sequence is characterized by an extraordinary degree of adaptability to perturbations, with the spider continuously integrating tactile feedback from each completed thread to adjust the geometry of subsequent silk deposition, producing webs of consistent functional quality even in physically challenging environments. The mechanical performance of spider webs under wind loading has attracted increasing attention from structural engineers as a model system for understanding how flexible lattice structures can be designed to absorb and distribute dynamic loads without catastrophic failure. Unlike rigid structures that resist loading by stiffness and tend to fail suddenly when forces exceed yield strength, spider webs respond to wind gusts through a combination of aerodynamic drag reduction. The silk threads and mesh geometry minimize surface area perpendicular to airflow at high tensions, and selective local failure that preserves the majority of the web's structural integrity. Research at MIT has demonstrated that orb webs exhibit the counterintuitive property of becoming stronger under the localized damage caused by prey impact or branch contact. because silk threads respond to failure of neighboring threads by stiffening through a molecular mechanism involving stress-induced crystallization of the amorphous regions of the silk polymer, redistributing load away from the damaged zone. The mathematical modeling of these stress distribution properties has directly influenced the design of engineered mesh structures in applications ranging from aerospace components to architectural facades, with spider silk serving as a biological existence proof for performance targets that conventional materials science had not previously achieved. The ongoing cross-pollination between arachnology and materials engineering represents one of the most productive interfaces between biological observation and technological innovation in contemporary science. The pirate spiders of the family Mimetidae represent an evolutionary strategy of predatory specialization so extreme as to constitute a remarkable example of parasitism within a predatory lineage. These spiders have abandoned the construction of prey capture webs entirely and instead specialize in invading the webs of other spider species and consuming the resident. Their technique involves entering a foreign web and plucking the silk threads in patterns that mimic the vibratory signals of struggling prey or courting males, luring the web's owner toward the source of the false signal before seizing and injecting venom at a vulnerable joint between leg segments, a precise anatomical targeting that delivers venom with maximum efficiency. What makes this predatory system particularly remarkable from an evolutionary perspective is the apparent coevolutionary dynamic between Mimetidae and their spider prey. The pirate spiders appear to specialize in the prey types that produce the most information-rich and stereotyped vibrations, particularly web-building spiders that are highly attentive to specific classes of vibratory signals within their webs. Some Mimetidae species have been documented imitating mating signals of specific spider species, attracting males seeking females, a form of sexual deception that extends chemical mimicry principles into the vibrational domain. The predatory versatility of Mimetidae makes them significant contributors to the complex web of interactions that regulate spider community structure in terrestrial ecosystems. The sheet web spiders of the family Linyphidae, the money spiders of European folk tradition and the dominant spider family at high altitudes and latitudes worldwide, construct some of the most ecologically significant webs in terrestrial ecosystems despite being individually among the smallest and most inconspicuous members of their order. Linyphiids collectively construct the delicate, dew-spangled horizontal sheet webs visible across grasslands on cool autumn mornings, a landscape-scale phenomenon created by the summation of millions of individual engineering efforts that together constitute a significant structural modification of the lower atmospheric boundary layer in many habitats. These webs function as intercepting platforms beneath a disorganizing array of trip wires that knock prey from flight paths, causing them to tumble onto the horizontal sheet, where the spider waits inverted beneath the sheet surface and seizes the struggling prey through the mesh from below. The money spider designation in European tradition reflects the folk belief that a money spider, most often in the Linyphidae family, landing on one's person presages an incoming financial windfall, a superstition sufficiently widespread across England and Ireland to appear in documentation from the 17th century onward. The ecological significance of Linyphiids extends far beyond folk mythology. Population density estimates in productive grasslands can exceed several million individuals per hectare, making them significant predators of small insects and potential influences on plant herbivore dynamics through the suppression of phytophagous insect populations. The crab spiders of the family Thomisidae have evolved a hunting strategy of exceptional elegance, ambush predation from floral surfaces, that places them at one of the most energetically productive nodes in terrestrial food webs, the interface between flowering plants and their pollinators. These spiders, characterized by their laterally flattened bodies, laterograde leg arrangement allowing sideways movement, and the ability of many species to alter their body coloration to match the flower upon which they sit, require no web and expend no energy in prey pursuit, simply sitting motionless on or within flowers and seizing pollinating insects that approach in search of nectar and pollen. The color change ability of genera like Misumena, the flower crab spider of North American and Eurasian meadows, is particularly remarkable among spiders, achieved through the movement of yellow pigments, omochromes, between inner and outer cell layers of the integument in a process requiring several days for full expression but allowing individuals to match backgrounds ranging from pure white to vivid yellow. The ecological impact of tomicity on pollinator populations has been the subject of considerable scientific debate, with some studies suggesting significant suppression of bee visitation rates to flowers occupied by crab spiders and others finding compensatory behavioral shifts in bees that include the ability to visually detect the spiders through UV-sensitive photoreceptors and avoid occupied flowers preferentially, an evolutionary outcome expected if bee predation pressure has been sufficiently intense and consistent to drive the evolution of active avoidance behavior. The evolutionary origins of the spider order have been reconstructed through a combination of molecular phylogenetics, comparative morphology, and the analysis of a fossil record that, while incomplete, extends back far enough to illuminate the major transitions in spider evolutionary history with remarkable clarity. The earliest unambiguous spider fossils, from Devonian deposits approximately 380 million years old, already possess the fundamental anatomical features characteristic of all living spiders: the fused prosoma and opistosoma connected by a narrow pedicel, chelicerae with associated venom glands, and spinnerets, indicating that the defining body plan of the order was established very early in its history. These ancient spiders are thought to have been terrestrial predators exploiting the rapidly expanding insect faunus of the early land ecosystems, and their silk, while not yet organized into elaborate aerial webs as in many living species, likely served functions of burrow lining, egg protection, and sensing substrate vibrations that remain fundamental to spider silk use across many living lineages today. The divergence between the two major living spider lineages, the Opisthothelii, containing the vast majority of living species, and the Mesothelii, a relict group represented today only by the family Lephistiidae of Southeast Asia, appears to have occurred early in spider evolution, with Mesothelii retaining several anatomical features considered ancestral, including externally visible segmentation of the abdomen and spinnerets in a medial rather than posterior position. The Mesothelii, represented in the modern fauna exclusively by the family Lephistiidae, with approximately 100 described species distributed across a restricted range in Southeast and East Asia, are frequently described as living fossils, a designation that is simultaneously illuminating and misleading as applied to any biological entity that has successfully persisted through hundreds of millions of years of environmental change. These spiders retain anatomical features absent in all other living spiders that were present in Carboniferous arachnids, most notably the externally visible segmentation of the opistosoma. In all other living spiders, this segmentation is concealed beneath a smooth, unsegmented cuticle and the medial positioning of the spinnerets, which are located beneath the opistosoma rather than at its posterior tip. Lephistiid spiders construct trapdoor burrows in forest soils, lining them with silk and extending radial silk trip lines across the soil surface around the burrow entrance, an architecture that appears remarkably similar to that preserved in some Carboniferous fossil deposits and suggests a deeply conservative ecology as well as morphology. The persistence of Lephistiidae in the specific refugial habitats of humid Southeast Asian forests, while mesothelid relatives disappeared from all other regions, likely reflects the particular stability of tropical Asian forest environments across geological time rather than any unique adaptive superiority of the Lephistiid body plan, though their survival for hundreds of millions of years speaks compellingly to the adequacy of their fundamental design. The remarkable convergent evolution of similar web architectures and hunting strategies in distantly related spider lineages represents one of the most powerful arguments for the adaptive nature of these behaviors and structures, demonstrating that the physical and ecological constraints of terrestrial predation drive independent evolutionary experiments towards similar engineering solutions. The orb web architecture, for example, long considered the defining innovation of the family Arachnidae and their relatives, is now known to have evolved independently at least twice, once in the clade containing Arachnidae, Tetragnathidae, and Nephilidae, and once in the distantly related family Euloboridae. The Euloborid orb webs are architecturally nearly indistinguishable from Araneoid orb webs at the macroscopic level, yet are constructed from chemically and mechanically distinct silk, Euloborids have lost the venom glands entirely and rely on elaborate wrapping with ultrafine cribellar silk to immobilize prey, demonstrating that convergent evolution can produce nearly identical functional outcomes through fundamentally different material pathways. Similarly, the funnel web architecture has evolved independently in multiple families including Agilenidae, Atypidae, and the unrelated Australian Hexathelidae. Trapdoor burrow construction appears in at least a dozen different families, and cryptic flower-dwelling ambush predation has arisen repeatedly in tomicids, some crab-like tetragnathids, and several other lineages. A pattern of convergence that speaks to the profound constraint exerted by ecological opportunity on the trajectories of spider evolution. The nervous system of spiders is organized in a manner that reflects the extreme reduction and concentration of neural tissue characteristic of small arthropods, yet achieves the processing capacity for sophisticated behaviors through architectural specializations that compensate impressively for the constraints imposed by limited neural volume. In most spiders, the brain, or more precisely the subesophageal ganglion complex, fills essentially the entire cephalothorax, leaving very little space for any other tissue, and in some of the smallest species the brain tissue actually extends into the leg bases to accommodate adequate neural volume within the prosoma. This spatial constraint has driven the evolution of extremely compact neural circuits in which individual neurons may serve multiple computational roles simultaneously, and in which the ratio of neural tissue per unit behavioral output is arguably among the highest in the animal kingdom. Neurobiological research on Argiope spiders and jumping salticids has revealed that the visual processing pathways of spiders achieve remarkable computational efficiency through a hierarchical organization in which the four pairs of eyes contribute different types of pre-processed information to higher integrative centers rather than raw pixel data, reducing the downstream processing burden through early feature extraction in a manner convergent with vertebrate retinal processing but achieved through entirely different developmental and structural mechanisms. The jumping spider brain in particular has attracted significant interest as a model system for understanding how complex visual behaviors can be generated by neural circuits of minimal physical scale. Spider immunity, the biological systems by which spiders defend against pathogens, parasites, and foreign particles, has received dramatically less scientific attention than the more accessible and medically relevant immune systems of vertebrates and insects, yet constitutes a fascinating example of how arthropod innate immunity can be elaborated to meet the specific ecological challenges of predatory arachnid lifestyles. Spiders lack the adaptive immune system that allows vertebrates to develop antigen-specific responses to novel pathogens, but possess a sophisticated innate immune system that includes cellular components, hemocytes that can engulf foreign particles through phagocytosis, and humoral components including antimicrobial peptides, phenyloxidase cascade-mediated melanization reactions, and protease inhibitors that limit pathogen-derived tissue damage. The hemolymph of spiders contains several classes of antimicrobial peptide that have attracted pharmaceutical research interest for their broad-spectrum activity against bacteria resistant to conventional antibiotics. Of particular ecological relevance is the suite of defensive responses spiders deploy against parasitoid wasps and flies, which represent significant natural mortality sources for many species. Some spiders detect the oviposition attempts of parasitoid wasps chemosensorially and respond with grooming behaviors that remove eggs before hatching, while the immune responses triggered by eggs that do successfully adhere to the opistosoma include melanization, encapsulation of the egg within a darkened protein-rich matrix, a response that succeeds in killing parasitoid eggs in some cases and is overcome by specialized counter-adaptations in others. The patadigua of Colombia, identified as the world's smallest known spider from specimens collected in wet leaf litter in the Serrania de Boto mountain range, embodies the extreme miniaturization end of the spider size spectrum in a manner that raises profound questions about the biological limits of functional complexity in nervous tissue, muscular architecture, and sensory system organization. Adult males of patadigua measure approximately 0.37 millimeters in body length, barely visible to the unaided human eye under good lighting conditions, yet contain within this vanishingly small volume all of the fundamental anatomical systems required for successful predation, digestion, silk production, locomotion, and reproduction. The miniaturization of nervous tissue to these scales creates specific challenges for neural computation, as individual neurons below a certain minimum size lose their capacity for saltatory conduction, the mechanism that allows large myelinated vertebrate nerve fibers to transmit signals efficiently over long distances. And the total neural cell count available for implementing complex behavioral circuits becomes extremely limited. Research on miniaturized spiders and other tiny arthropods suggests that the neural circuits underlying basic predatory behaviors can be implemented with remarkably few neurons, but that more complex computationally demanding behaviors such as web construction are correspondingly simplified or absent in the most miniaturized species, suggesting that brain scaling places fundamental constraints on behavioral complexity at the lower extreme of the spider size distribution. The ecological impact of spiders on global insect populations constitutes one of the most significant yet least appreciated regulatory effects exerted by any predator group on terrestrial ecosystems, with annual global spider predation estimated to remove prey biomass on a scale that exceeds the total annual meat consumption of all 7 billion human beings combined. This staggering figure, derived from a synthesis of studies estimating spider density, individual prey consumption rates, and active predation periods across major biome types, reflects the extraordinary ubiquity of spiders in virtually every terrestrial habitat from Arctic tundra to tropical rainforest, their high metabolic efficiency relative to warm-blooded predators, and the disproportionate representation of small arthropods, particularly insects and collembolans, among their prey. Forest spider communities are estimated to consume between 1 and 4 tons of prey per hectare per year in productive temperate and tropical forests, while grassland communities achieve comparable or higher rates per unit area in agriculturally productive zones. The practical implications of this predation pressure extend into agricultural systems, where spider communities, even in intensively managed monoculture environments, exert measurable suppressive effects on pest insect populations, with some studies demonstrating that removal of natural enemy populations including spiders increases pest-mediated crop losses by factors of 2 to 3 in the absence of pesticide applications. The conservation of spider communities in agroecosystems thus represents a potentially significant component of sustainable pest management strategies that remains underutilized in most global agricultural frameworks. The discovery of spider silk's potential as a medical biomaterial has catalyzed an explosion of biotechnological research over the past 3 decades, driven by the recognition that spider silk's unique combination of mechanical properties, biocompatibility, and biodegradability positions it as an ideal candidate for applications ranging from suture materials and wound dressings to tendon and ligament repair scaffolds, drug delivery matrices, and even elements of neural interface technology. The central challenge limiting the medical application of spider silk is the same challenge that prevented its industrial exploitation for centuries. The territorial and sometimes cannibalistic nature of spiders makes farming them for silk production economically impractical, requiring that the silk proteins be produced through biotechnological means. Recombinant spider silk proteins have been successfully expressed in bacterial systems, yeast, mammalian cell lines, plant tissues, and transgenic silkworms and goats, with each expression system producing somewhat different protein variants due to the challenges of replicating the full-length spidroin gene sequences with their extensive repetitive domains in foreign cellular environments. The startup company Bolt Threads and the research spin-out AM Silk have both developed commercial-scale recombinant spider silk production systems, producing materials with properties approaching but not yet fully matching native spider silk, and have achieved regulatory approval for several cosmetic and some medical applications. The gap between recombinant and native silk properties continues to narrow as researchers develop more sophisticated understanding of the spinning process. The liquid crystal prealignment of silk proteins in the spinning dope and the flow-induced crystallization during drawdown through the spigot, and apply this understanding to biomimetic spinning systems. The spinnerets through which spider silk is externalized are themselves organs of remarkable mechanical sophistication, representing not merely exit orifices for preformed silk but active participants in the silk drawing process that critically determine the final mechanical properties of the extruded thread. Each spinneret contains multiple spigots, the individual silk-releasing nozzles, each connected to a specific class of silk gland, and the coordinated output of these spigots can be combined into composite silk structures of varying composition and architecture depending on the spider's behavioral state. The drawing velocity imposed on the silk as it exits the spigot, controlled by the spider's leg movements and behavioral decisions about how rapidly to move while releasing silk, critically determines the degree of molecular orientation and crystallization in the final fiber, with faster drawing velocities producing stronger, stiffer fibers and slower drawing producing more extensible, elastic threads. This remarkable capacity for real-time mechanical property tuning through behavioral adjustment of drawing speed allows individual spiders to produce silk of different mechanical characters for different functional contexts without changing the molecular composition of the spinning dope, a form of mechanical processing sophistication that has inspired the design of variable property synthetic fiber extrusion systems in polymer engineering. The spinneret apparatus also includes complex valve mechanisms that allow the spider to start and stop silk flow from individual spigots with great precision, enabling the construction of composite silk structures with controlled layering of different fiber types. The phenomenon of kleptoparasitism in spiders, the theft of prey or silk resources from the webs of other spiders, has been documented in numerous lineages and represents an evolved behavioral strategy that exploits the productive infrastructure created by web-building species at the expense of their energetic investment. The silver garden spider, Argiope argentata, is frequently targeted by the kleptoparasitic dewdrop spider, Argyrodes elevatus, a tiny theridiid that lives within the larger spider's web, stealing small prey items and consuming web silk directly as a nutrient source, behaviors that have evolved alongside behavioral adaptations to avoid detection and predation by the host. The ecological relationship between kleptoparasitic Argyrodes species and their host spiders is one of the best studied examples of parasitism within the spider order, revealing a dynamic in which host spiders tolerate low densities of kleptoparasites while actively attacking and evicting individuals that become too numerous or whose thefts exceed a behavioral threshold, suggesting cost-benefit calculations operating in the host's tolerance decisions. The body size of kleptoparasitic spiders is characteristically very small relative to their hosts, a morphological adaptation that minimizes the metabolic cost of maintaining residence in a foreign web while reducing the prey size that can be stolen, limiting the per-individual cost to the host and thus extending the range of densities that hosts tolerate before retaliating. Some Argyrodes species have been documented modifying the web architecture of their hosts by systematically removing threads, an intervention that has been interpreted as facilitating the kleptoparasite's movement through the web while potentially degrading its prey-catching performance. The orb-weaving spider Nephila, now formally reorganized taxonomically into the genus Trichonephila following molecular phylogenetic revision, produces what is arguably the finest and most extensively studied silk of any spider genus, and the extraordinary engineering of Nephila webs has served as the primary model system for understanding orb web biomechanics, silk material science, and the sensory ecology of web-based predation across several decades of arachnological research. The golden silk of Nephila, the source of the common name golden silk orb-weaver, is yellow in color due to the incorporation of specific flavonoid-like pigments whose functional significance is debated, with proposed roles ranging from camouflage against sunlit backgrounds to UV attraction of prey to potential antimicrobial properties. The scale of Nephila web structures in tropical environments is remarkable. In the forests of Madagascar and Queensland, individual Nephila innerata and Nephila idulis webs have been measured with frame circumferences exceeding 1.5 meters and radii of over 30 centimeters, capable of intercepting prey as large as small bats and birds, whose occasional entanglement in large Nephila webs has been photographically documented. The sexual dimorphism of Nephila is among the most extreme in the spider order, with females achieving body lengths of 4 to 5 centimeters while dwarf males measure merely 4 to 6 millimeters, a size difference of over 10-fold that is thought to reflect extreme selection on female body size for reproductive output combined with selection on male miniaturization to reduce the risk of predation by the female. The spiders of Madagascar represent a microcosm of the broader evolutionary patterns that make island biotas such uniquely valuable systems for understanding evolutionary diversification, ecological specialization, and the interplay between geographic isolation and biological innovation. Madagascar's extraordinary biological heritage, including endemic spider fauna of remarkable diversity and uniqueness, reflects its long isolation from African and Asian landmasses following the breakup of Gondwana, allowing lineages that arrived by overwater dispersal to diversify in ecological isolation over tens of millions of years. Among the most remarkable of Madagascar's endemic spider species is Carostris darwini, the Darwin's bark spider, named in honor of the naturalist, which produces a web silk of extraordinary toughness that was, when first formally measured and reported in 2010, the toughest biological material ever recorded, exceeding by more than an order of magnitude the toughness of ordinary spider silk and approximately 10 times that of the Kevlar used in ballistic protection. Darwin's bark spider constructs its webs spanning rivers and lakes, sometimes across water surfaces exceeding 25 meters in width, using anchor lines of silk that have been measured at individual lengths of up to 25 meters, the longest silk lines recorded for any spider species. These extraordinary web dimensions and the remarkable mechanical properties required to maintain structural integrity in silk anchor lines of such length under real-world loading conditions represent an evolutionary achievement that continues to challenge materials scientists' models of biological fiber performance. The peacock spiders of Australia, a spectacularly diverse radiation within the jumping spider genus Moratus, with over 90 described species and likely many more awaiting formal description, have attracted extraordinary popular and scientific attention since high-quality photographic documentation of their courtship displays became widely circulated in the early 2010s, revealing a world of miniaturized visual spectacle that had been hidden in plain sight beneath the feet of hikers and naturalists across the Australian continent for generations. Male Moratus spiders possess brilliantly colored abdominal flaps that can be raised during courtship into iridescent semicircular fans displaying patterns of extraordinary complexity and color saturation, rivaling in their visual impact the elaborate tail displays of birds of paradise despite being produced by an animal with a body length typically less than 5 millimeters. Each species possesses a distinct display pattern that differs from relatives in coloration, geometry, and the behavioral choreography through which it is presented to attending females, suggesting rapid speciation driven by female choice in a manner paralleling the runaway sexual selection dynamics proposed to explain the extreme ornamentation of bird of paradise males. The optical mechanisms producing the brilliant colors of Moratus displays involve not merely pigmentation but structural coloration, the interference of light waves scattered from nanoscale physical structures in the integument that produce iridescent hues far more saturated and spectrally pure than chemical pigments can achieve, a convergent evolution of structural coloration with birds, butterflies, and beetles that represents a fascinating example of similar physical solutions emerging across vastly different taxonomic contexts. The wolf spiders of the family Lycosidae, one of the largest spider families with over 2,400 described species distributed globally across virtually every terrestrial biome, represent the dominant hunting strategy among cursorial spider predators, pursuing prey actively across substrate surfaces rather than relying on web architecture or ambush from concealment. Their common name acknowledges this pursuit predation style, and their anatomical organization reflects the biomechanical requirements of cursorial hunting: strong, well-muscled legs capable of sustained rapid locomotion, 8 eyes arranged in 3 rows with the large secondary eyes of the middle row providing wide-field motion detection, and a robust prosoma that anchors powerful chelicerae musculature for securing and subduing struggling prey. The maternal care behaviors of lycosid spiders are among the most elaborate in the order, with females carrying their egg sac attached to the spinnerets rather than leaving it in a protected retreat, then, after hatching, carrying the spiderlings on their back for a period of days to weeks before the young disperse. This extended maternal care involves not merely passive transport but active behavioral responses to threat, with mothers turning to face disturbances and displaying defensive postures while carrying offspring, and spiderlings responding to threats by clustering more tightly on the maternal opisthosoma rather than dispersing, a coordinated anti-predator response that suggests chemical and vibratory communication between mother and offspring during the carrying period. The fascinating world of chemosensory communication in spiders has emerged as one of the most productive research frontiers in arachnology over the past 2 decades, revealing a rich chemical language mediated by contact pheromones, volatile signals, and substrate-borne chemical traces that governs mate finding, species recognition, prey detection, rival assessment, and predator avoidance across the order. Spiders possess chemical receptors distributed across the tarsi, the terminal leg segments, that make contact with the substrate as the spider walks, enabling continuous chemosensory sampling of chemical traces left by previous occupants of that surface. The chemical composition of spider cuticle, the outermost layer of the exoskeleton, has been revealed through gas chromatography mass spectrometry analyses to contain dozens to hundreds of cuticular hydrocarbons and other volatile compounds in species-specific, sex-specific, and age-specific patterns that serve as contact pheromones mediating multiple aspects of social interaction. Research on Tegenaria spiders, the house spiders familiar to European residents, demonstrated that female cuticular chemicals provide males with information sufficient to discriminate between virgin and mated females, between females of different species, and potentially between females of different genetic quality, allowing selective mating decisions based on chemosensory assessment rather than visual or vibratory information. The substrate-borne silk itself carries chemical information, with silk draglines deposited by female spiders containing chemical signals that attract and orient searching males over distances extending far beyond direct contact range. The extraordinary feat of prey capture achieved by the net-casting spider Dinopus, sometimes called the gladiator spider for the posture it adopts when deploying its hackled band web, involves a projectile net throw of such speed and precision that high-speed video analysis was required to reveal its kinematic details, demonstrating that the spider can accelerate its front legs at rates exceeding 4,000 meters per second squared during the strike, an acceleration comparable to that experienced during artillery shell discharge. The hackled band's silk constituting the diinipid net is qualitatively different from all other known spider silks, being produced by the cribellum, a modified spinneret composed of thousands of microscopic spigots, and combed into a woolly, three-dimensional mesh by the calamistrum, a row of curved setae on the rear legs. The resulting cribellar silk consists of nanoscale threads wound into a spiral nanostructure around core supporting fibers, creating a material with an extraordinary surface area per unit mass that adheres to prey through van der Waals forces and physical entanglement rather than chemical stickiness, making it effective against both smooth and hairy insect surfaces. The net is stretched between the spider's front legs at 4 to 6 times its resting dimensions before deployment, storing elastic strain energy that is released explosively during the strike, a biological spring mechanism that allows the spider to achieve the millisecond scale throwing speeds revealed by high-speed imaging. The phenomenon of prey size range extension through cooperative prey capture in social spiders represents one of the most dramatic examples of how cooperative behavior can fundamentally alter the ecological role of a species in its ecosystem, enabling social spider colonies to capture prey orders of magnitude larger than any individual could manage, thereby occupying a predator niche inaccessible to solitary spider species of similar individual body size. Research on the neotropical social spider Anolosimus eximius demonstrated that large colonies can capture vertebrate prey including hummingbirds, frogs, and small lizards in cooperative attacks involving dozens of colony members simultaneously attacking and enveloping the prey items that would be entirely invulnerable to single individuals of the same species. The coordination of these multi-individual attacks does not appear to involve sophisticated communication or leadership but rather emerges from simple stimulus-response rules in which each individual responds to the vibrations and chemical signals produced by struggling prey and by the attack behaviors of neighboring colony members. creating a positive feedback dynamic that recruits progressively more attackers until the prey is overwhelmed. This emergent collective predation demonstrates how individually simple behavioral rules can generate complex, functional group behaviors, a principle with broad theoretical implications for understanding the evolution of collective intelligence in social animal systems without requiring the attribution of central coordination or planning to any individual actor. The silk of the cribolate spiders, those possessing the primitive silk-producing organ, the cribellum, from which hackled band silk is extruded, has been the subject of increasing materials science research since the recognition that its adhesion mechanism, based on dry contact forces rather than chemical wetness, represents a potential model for the development of synthetic dry adhesives with properties unachievable through conventional pressure-sensitive adhesive chemistry. Cribolate silk adheres through a combination of van der Waals interactions between the nanoscale looped fibers of the woolly silk mesh and the substrate surface, and through mechanical interlocking of these fibers with surface asperities and hairs at the micro and nanoscale. This adhesion mechanism is directional in many systems, providing strong resistance to pull off in one direction while allowing relatively easy sliding in another, a property that mirrors the directional adhesion of geckosidae and is similarly attractive for engineering applications requiring controlled release, reusable adhesion. The biological elegance of cribellar's silk adhesion lies in its robustness across diverse surface types. Unlike chemical adhesives that may fail on contaminated or unusual surfaces, and unlike smooth surface van der Waals adhesives like gecko-inspired materials that require specific surface geometry for effective contact, the high surface area and compliance of cribellar silk allows effective adhesion to surfaces ranging from smooth glass to rough bark to the setose cuticle of insect prey. This versatility makes cribellar's silk a particularly compelling biological model for the engineering design of robust, context-independent adhesive systems. The remarkable long-term population dynamics of spider communities in stable ecosystems have been illuminated through several decades-long monitoring studies that reveal boom and bust cycles, community succession patterns, and responses to climate variation that challenge simplified conceptions of spider populations as static background elements of terrestrial invertebrate communities. Studies in British heathlands, Swiss alpine meadows, and North American tallgrass prairies have documented multi-year population cycles in dominant spider species that correlate with prey availability, microhabitat structure, and weather patterns in ways that suggest complex indirect interactions through shared prey populations, apparent competition, and intraguild predation among spider species. Climate warming has emerged as a significant driver of spider community change across multiple study systems, with range expansions by southern species into formerly marginal habitats, phenological advances in breeding and emergence, and altered community composition reflecting differential responses to warming by species adapted to different thermal regimes. Critically, the disproportionate temperature sensitivity of small ectotherms relative to larger homeothermic species means that spider communities may serve as particularly sensitive biological monitors of microclimate change, with the species composition and activity patterns of local spider assemblages potentially providing earlier and more detailed signals of climate trajectory than many other ecological indicators. The integration of spider community monitoring into long-term ecological research programs thus represents an underutilized opportunity for both basic ecological understanding and applied climate change assessment. The genus Latrodectus, encompassing the widow spiders in all their geographic diversity, has a distribution that reflects both ancient biogeographic history and more recent human-mediated dispersal, with species present on every inhabited continent and on many remote islands. A cosmopolitan pattern unusual among specialist predators and reflecting the ability of widow spiders to exploit the human-modified environments, storage spaces, agricultural buildings, ports, and shipping containers that have connected the globe through commerce. The remarkable pharmacological complexity of Latrodectus venom, which varies not merely between species but between populations of the same species and between individual spiders as a function of sex, age, and dietary history, has made it a productive system for neuropharmacological research, with latrotoxin family members serving as precise molecular tools for dissecting the mechanisms of neurotransmitter release at synapses. Beyond alpha-latrotoxin, which targets vertebrate neurons, Latrodectus venoms contain insect-specific and crustacean-specific toxins, alpha-latroinsectitoxin and alpha-latrocrustatoxin, with distinct receptor targets that are not expressed in vertebrate nervous systems. An observation that suggests the venom has been shaped by selection involving both prey capture and defensive functions, and that has informed the development of taxonomically selective insecticides. The silk of Latrodectus has been described as among the mechanically strongest of all spider silks, with the structural framework of cobwebs, particularly the strong, non-sticky scaffolding threads, exhibiting tensile properties approaching those of major ampullate gland silks from orb-weavers, though their irregular architecture distributes loads differently from the geometric precision of orb webs. The philosophical and cultural resonance of the spider's web as a symbol of connectivity, fragility, impermanence, and hidden structure has made it one of the most pervasive metaphors in human intellectual and artistic tradition, appearing across domains from theology to political philosophy to computer science in ways that reveal the web's power as a conceptual model for the structure of complex relationships. In Indra's net of Vedic and later Buddhist cosmology, the universe is imagined as an infinite net stretched across space with a jewel at each node, each jewel reflecting all other jewels in an endlessly recursive pattern of mutual interdependence. A cosmological model that preceded by millennia the network topology concepts now fundamental to information science and graph theory. The appropriation of web imagery in early descriptions of the World Wide Web was not coincidental. The vocabulary of the web, nodes, links, networks, spiders, still the name for information indexing programs, reflects a genuine conceptual debt to the spatial and relational logic that spider webs embody physically. Political theorists from Machiavelli forward have employed web metaphors to describe the invisible structures of power and influence through which ruling entities maintain control, a conceptual use of the spider's unseen architecture as a model for institutional invisibility that persists in contemporary political discourse. The web's simultaneous qualities of incredible strength and extreme fragility, shattering under the mass of a raindrop yet supporting prey many times the spider's own weight, have made it an enduring symbol for the paradoxical coexistence of vulnerability and resilience in human social and natural systems. The physiological mechanisms underlying the molting process in spiders, the periodic shedding of the exoskeleton that allows growth to proceed in an animal enclosed within a rigid chitinous shell, constitute one of the most physiologically demanding events in the life of any arachnid, requiring coordination of hormonal signals, enzymatic tissue remodeling, behavioral preparation, and a precisely timed biomechanical sequence of escape from the old cuticle that must be completed within a relatively narrow time window or risk fatal entrapment. Molting in spiders is controlled by ecdysone hormones, as in insects and other arthropods, which trigger the separation of the old cuticle from the underlying epidermal cells, the synthesis and partial hardening of the new cuticle beneath the old, and the enzymatic dissolution of the innermost layers of the old cuticle to allow its detachment. The molt itself begins with a split along the anterior margin of the opistosoma, through which the spider extracts first its prosoma and then, with sustained muscular effort, each of its legs, a process that requires the legs to be pulled free of their old casings while the new exoskeleton beneath is still soft enough to fold and compress. The vulnerability of spiders during and immediately after molting, when they are effectively immobile with a soft new cuticle that will not harden for hours or days, is a significant source of natural mortality, and most spiders prepare carefully for molts by constructing a sheltering molting hammock of silk, often in a concealed location, and ceasing to eat for several days beforehand to reduce abdominal volume and facilitate extrication from the old exoskeleton. The evolution of the pedicel, the narrow waist connecting prosoma and opistosoma in all spiders except the primitive mesotheli, represents a morphological innovation of profound functional significance whose implications for spider biology extend across locomotion, silk production, sensory physiology, and the mechanics of mating in ways that illuminate why this structural feature was retained and elaborated through hundreds of millions of years of spider evolution. The pedicel consists of an extremely narrow ring of cuticle and soft tissue that allows the opisthossoma to rotate independently of the prosoma, enabling the spinnerets to be aimed in three-dimensional space without requiring the spider to reposition its entire body, and simultaneously transmits the nerve cords, major blood vessel, digestive tube, and silk gland ducts between the two body regions through an architecture of extraordinary compactness. The mechanical flexibility of the pedicel allows spiders to place silk in precise spatial orientations during web construction, dramatically expanding the architectural possibilities available to web-building species compared to what would be achievable if the opisthossoma were rigidly fused to the prosoma as in their chelicerae relatives. The pedicel also functions as a load-bearing element of considerable sophistication. During prey struggling events, the pedicel transmits the forces generated by the opisthosoma's mass swinging in response to web vibrations while simultaneously protecting the delicate neural and circulatory connections from tensile failure, a biomechanical requirement that has shaped the microarchitecture of pedicel cuticle toward particularly high tensile strength and toughness. The remarkable recent discovery of spider species in habitats previously considered entirely inhospitable to arachnid life has continuously revised our understanding of the physiological limits of the spider body plan and the extremes of environmental tolerance achievable within a fundamentally ectothermic arthropod physiology. Species of the family Nephosidae and Linyphidae have been documented active on the ice surface of glaciers in the European Alps and the Himalayas, hunting collembolans and other small invertebrates blown onto the ice surface by alpine winds. An ecological lifestyle requiring physiological cold tolerance far beyond what is achievable by most invertebrate ectotherms and involving antifreeze mechanisms including elevated trehalose and other cryoprotectant concentrations in the hemolymph. At the opposite thermal extreme, certain desert-adapted spider species in the Namib and Saharan deserts tolerate body temperatures exceeding 40 degrees Celsius during midday activity, employing behavioral thermoregulation by stilting, elevating the body on extended legs to place it in the cooler boundary layer above the hot sand surface. And stilting efficiency has been demonstrated to increase with leg length in a manner suggesting ongoing selection for longer legs as a thermoregulatory adaptation in hot desert environments. Intertidal spiders, species from several families that occupy the splash zone and even the intertidal zone of rocky coastlines, tolerate periodic immersion in seawater through a combination of behavioral air bubble trapping, respiratory shutdown, and osmoregulatory mechanisms whose molecular details represent a virtually unexplored frontier of comparative arachnid physiology. The extraordinary diversity of prey capture strategies represented within the single family Theridiosomatidae, the ray spiders, provides a particularly vivid illustration of how evolutionary specialization can produce radical functional divergence from a conservative ancestral body plan through the modification of web architecture and silk handling behavior rather than through obvious morphological change. The ray spiders build a distinctive three-dimensional dome-shaped web under tension by attaching a central thread to the hub that, when released, springs the web forward with remarkable speed, a mechanism that has evolved from the simpler cone-shaped sheet web of ancestral Theridiosomatids through progressive elaboration of the spring-loading mechanism. Research by William Eberhard at the Smithsonian Tropical Research Institute revealed that this spring-loading behavior serves as an active prey capture mechanism. The spider holds the tension thread taut with its front legs while waiting, and upon detecting the approach of a flying insect, releases the thread, causing the entire web to spring forward and entangle the prey through a directed mechanical strike rather than passive interception. The biomechanical analysis of this behavior, requiring precise timing, directional release, and a web architecture specifically optimized for rapid spring loading and efficient energy transfer to the striking face, represents a sophisticated solution to the engineering challenge of capturing fast-flying prey with a structure that must simultaneously serve as a passive prey intercept web when the spider is not actively deploying it. The prey capture success rates of spring loading versus passive interception in these spiders have been compared experimentally, confirming that the active strike provides measurable advantages for capturing the small dipterans and other flies that constitute the primary prey. The mythological and symbolic treatment of the spider in the literary tradition of the European Middle Ages and Renaissance was far more nuanced and less uniformly negative than a superficial reading of the historical record might suggest, with spider imagery deployed across a wide spectrum of symbolic registers from the diabolical to the divine, the industrious to the malicious, in ways that reflected both classical precedents and original theological and moral elaboration. The Psalter commentary tradition in medieval Christianity elaborated spider imagery from passages in the Book of Job and the Psalms, where the spider's web appears as a symbol of the transience of human ambition and the fragility of earthly construction, into extended allegorical readings in which the spider's patient predatory waiting became a figure for the devil's ambushing of sinful souls, her web of sin entrapping the spiritually careless. Yet simultaneously, other medieval commentators celebrated the spider as a figure of ingenious industry and providential design, the precision of her web-building read as evidence of divine mathematical order embedded in nature, her silk as a marvel of creation that human art could not approach. Francis Bacon's influential 1620 metaphor contrasting the spider's spinning from her own substance with the ants merely collecting and the bees ideally transforming external materials into something new provided a framework for epistemological discourse about the proper method of natural inquiry that shaped scientific philosophy for generations, demonstrating how spider behavior could become a vehicle for entirely non-biological intellectual debates. The capacity of spiders to produce multiple, chemically distinct silk types from a single organism represents a striking contrast with the silk production systems of other silk-producing arthropods, most notably the silkworm Bombyx mori, whose cocoon silk has supported an industry of enormous cultural and economic significance across East Asia and the Mediterranean for millennia but is biochemically a much simpler product. Where silkworm silk consists essentially of two proteins, fibroin and sericin, produced by two gland types. The silk gland complement of an advanced orb-weaving spider may include 7 or more gland types producing biochemically distinct spidroins with non-overlapping mechanical functions. This biological complexity of spider silk production has made spiders a comparatively intractable subject for silk industrialization despite the superior material properties of spider dragline silk relative to silkworm cocoon silk, driving the sustained investment in biotechnological alternatives that has characterized the spider silk materials industry over the past 3 decades. The silkworm itself has been the subject of genetic engineering efforts to produce spider silk proteins, with transgenic Bombyx mori lines expressing Nephila dragline silk genes producing cocoon fibers with significantly enhanced toughness relative to normal silkworm silk, demonstrating the feasibility of using established sericulture infrastructure to produce spider silk-based materials if the biological production challenges can be adequately solved, though matching the full mechanical complexity of native spider silk through biological expression in a foreign organism remains a challenging goal. The neurotoxicological mechanisms of spider venoms acting on voltage-gated sodium channels represent one of the most intensively studied systems in ion channel pharmacology, with spider toxins providing molecular tools of extraordinary specificity that have enabled the dissection of channel gating mechanisms with a precision unavailable through conventional electrophysiological approaches alone. Voltage-gated sodium channels are the primary drivers of action potential generation and propagation in the nervous systems of essentially all animals, and their modulation by specific toxins allows precise manipulation of electrical excitability in neurons and muscle fibers with which the pharmacological effects of natural selection have had tens of millions of years to optimize. The delta-atricotoxins of atrax and related Australian funnel web spiders, the huan toxins of the Chinese bird spider Cereopagopus schmidti, and the jingza toxins of the Chinese earth tiger tarantula act on sodium channels through distinct binding mechanisms that stabilize different functional states of the channel and produce correspondingly distinct pharmacological effects ranging from repetitive firing to persistent channel opening. Structural studies resolving the three-dimensional architecture of spider toxin sodium channel complexes at near-atomic resolution through cryo-electron microscopy have recently illuminated the molecular basis of toxin selectivity across the 9 sodium channel subtypes expressed in human tissues, a fundamental advance that directly informs structure-guided drug design efforts targeting pain, epilepsy, and cardiac arrhythmia through selective modulation of specific sodium channel subtypes. Implicated in these conditions, the biogeographic patterns of spider diversity across the globe reflect the interplay of continental drift history, climate history, colonization ability, and the evolutionary biology of individual spider lineages in ways that have only begun to be systematically analyzed through the integration of molecular phylogenetics with paleoclimatic and paleontological data. The highest spider species richness on Earth is concentrated in humid tropical regions, particularly in the Indo-Pacific region encompassing Southeast Asia, the Indonesian archipelago, and the Pacific island chains, where the combination of ancient landmass stability, climate stability through geological time, and complex habitat heterogeneity has allowed the accumulation of extraordinary diversity through in situ diversification and over-water dispersal from multiple source regions. Australia and New Zealand represent particularly interesting biogeographic case studies for spiders, as they were isolated from other major landmasses at different times during Gondwana fragmentation and received different subsequent colonization histories, yet both support spider faunas of remarkable endemism, with Australia's spider fauna roughly 90% endemic at the species level, that reflect the evolutionary potential of geographically isolated lineages diversifying in the absence of major immigration. The dispersal ability that ballooning provides to small spider species creates interesting tensions in biogeographic interpretation, as even apparently strongly endemic lineages may include elements of recent long-distance immigration that confound phylogeographic reconstructions based on the assumption of geographic isolation and in situ diversification. The phenomenon of thanatosis, feigning death, has been documented in numerous spider species across multiple families and represents a defensive behavioral strategy of considerable subtlety that exploits the tendency of many predators to discriminate against non-moving prey items as potentially spoiled or dangerous. When threatened by a predator, a spider displaying thanatosis will abruptly go limp, pulling its legs against its body in a characteristic flexed posture and remaining completely motionless for periods ranging from seconds to many minutes, depending on the species and the persistence of the threat stimulus. The behavioral decision to enter and maintain thanatosis, and the duration of the unresponsive period, appear to be influenced by the predator's behavior, with continued prodding or sniffing by the predator typically extending the duration of immobility, suggesting that the spider continues to process sensory information during the feigned death state and modulates its behavior accordingly, rather than entering a completely inactivated or reflexive state. The evolutionary maintenance of thanatosis as a defensive strategy requires that predators sufficiently often reject motionless prey to provide a fitness advantage over alternative defenses such as flight, biting, or venom injection, and documented cases of spider-eating predators including wasps, lizards, and birds accepting only moving prey while rejecting motionless individuals of identical apparent quality demonstrate that this condition is often met in nature. The species specificity of thanatosis postures and durations suggests that this behavior, like other anti-predator adaptations, is subject to ongoing selection from specific predator assemblages in each species range. The Phalcidae, the cellar spiders or daddy long-legged spiders, represent one of the most ecologically successful spider families in human-modified environments, having colonized the built environment so thoroughly that many of the approximately 1,900 described species are now found predominantly in human structures across regions far outside their natural ranges. The elongated, delicate leg architecture that gives phalcids their evocative common name serves multiple functional roles, including long-reaching web manipulation for prey and egg sac handling, the production of long-range vibratory signals through a behavior called bouncing, rhythmic oscillation of the body within the web that has been interpreted as prey mimicry, courtship signal, and anti-predator confusion behavior in different contexts, and potentially a role in increasing effective body size during threat displays. A widely circulated folk belief maintains that phalcid spiders are the most venomous of all spiders but are prevented from harming humans only by the inadequate size of their chelicerae, a claim that has been definitively and reproducibly falsified by both cheliceral morphological measurements demonstrating that phalcid fangs can and do penetrate human skin during deliberate contact, and by venom analyses showing that falsid venoms, while effective against prey and against other spiders, have no exceptional potency against vertebrate tissues. Falcidae are significant intragilled predators in human structures, commonly consuming other spiders including small web-building theridiids and even young house spiders, making them net contributors to the reduction of total spider diversity in buildings despite their own high abundance. The remarkable behavioral flexibility demonstrated by some spider species in their responses to changing environmental conditions has challenged the traditional view of spiders as largely stereotyped, reflexively programmed predators incapable of meaningful behavioral modification through experience. Research on orb-weaving spiders including Lirynioids Sclopitarius and Zygiella exnotata has demonstrated that individual spiders modify their web architecture in response to past prey capture success in ways consistent with learning or adaptive behavioral adjustment, investing more heavily in web structures that previously produced prey captures and reducing investment in structures that have consistently yielded poor returns. The capacity for spatial learning in the wolf spider Lycosa tarantula, the spider that lent its name to the supposedly spider bite-induced dancing mania of medieval southern Italy, and whose association with the tarantella folk dance constitutes one of the most fascinating intersections of arachnology and medical history, has been demonstrated through maze navigation experiments in which individual spiders reliably improved their performance across repeated trials, suggesting short-term spatial memory of at least several minutes duration. The implications of these findings extend beyond individual behavioral ecology into the broader theoretical question of the neural architecture required for learning, as the demonstration of experience-dependent behavior modification in animals with such miniaturized nervous systems places important constraints on models claiming that learning requires specific neuroanatomical structures or minimum neural circuit complexity unavailable to small invertebrates. The human fear of spiders, arachnophobia, is among the most common specific phobias in human populations worldwide, consistently reported at clinical diagnostic levels in between 3 and 6% of surveyed populations, with subclinical spider aversion considerably more widespread, and its evolutionary origins have been the subject of productive scientific debate that illuminates broader questions about the architecture of evolved emotional responses to categories of environmental stimuli. The evolutionary psychology hypothesis that arachnophobia reflects a pre-adapted fear response to ancestrally dangerous spiders that provided survival advantages to hominins in environments where venomous spider encounters carried meaningful mortality risk, receives some support from developmental data showing that human infants and young children show enhanced attentional capture and more rapid threat responses to spider images compared to size-matched control images, even in populations with no prior experience of spider encounters. Cross-cultural data complicates simple evolutionary accounts, however, as arachnophobia rates vary substantially between cultures in ways that correlate with cultural attitudes towards spiders, suggesting a significant learned component that interacts with any innate attentional bias. Neuroimaging research has revealed that spider images activate the amygdala, the brain structure associated with threat detection and fear conditioning, with notably greater speed and lower threshold than most other threat stimuli, suggesting that spiders may constitute a biologically prepared stimulus category for which the fear response circuit has an evolutionarily reduced activation threshold, without implying that the phobia itself is genetically fixed or unavoidable. The spiders of the genus Portia, widely distributed across sub-Saharan Africa, Southeast Asia, and Australia, have become the most extensively studied subjects in cognitive arachnology and represent the strongest existing evidence that complex problem-solving behaviors, including what appears to be planning and insight-based reasoning, can occur in animals with nervous systems of extreme miniaturization. Portia species are araneophagic jumping spiders that specialize in hunting other spiders by exploiting their vibratory communication systems, plucking web threads in patterns that mimic prey, rival spiders, or courting mates, and adjusting these deceptive signals based on real-time behavioral feedback from the resident spider, a form of trial-and-error problem-solving that requires the integration of sensory input with a flexible behavioral repertoire in ways that appear to exceed the capacity of simple reflex-mediated systems. The detour experiments of Fiona Cross and Robert Jackson, in which Portia fimbriata navigated three-dimensional laboratory structures to reach prey visible only from specific vantage points, required the spiders to initially move away from the prey, a counterintuitive path component inconsistent with simple gradient-following strategies, and to maintain a representation of the prey's location throughout a navigation sequence taking many minutes. The neural substrate enabling this apparent planning capacity remains poorly understood, as the Portia brain contains only a few 100,000 neurons, orders of magnitude fewer than the simplest vertebrate brain, challenging neuroscientists to consider whether the computational principles underlying planning and problem-solving might be implementable in neural circuits far simpler than previously imagined possible. The spider's role in human material culture, extending far beyond the legendary silk industries that failed to take root due to production difficulties, encompasses a surprisingly rich history of direct silk utilization for practical purposes across cultures that found ways to exploit this remarkable material despite the challenges of farming its producers. Indigenous cultures in Papua New Guinea and the Solomon Islands traditionally harvested the massive webs of Nephila spiders stretched across bamboo frames to create fishing lines and bags of remarkable strength relative to their weight, a practice that represents a direct material use of spider silk without any processing beyond collection and shaping. The reticle crosshairs in optical instruments including rifle scopes, surveying theodolites, and astronomical telescopes were manufactured from spider silk for over 2 centuries before synthetic alternatives became available, with the silk preferred for its extreme fineness, uniformity, and mechanical stability across temperature and humidity ranges. The earliest authenticated use of spider silk in European material culture appears in records of the French naturalist Bon de Saint-Hilaire, who in 1709 demonstrated silk stockings and gloves manufactured from the silk of egg sacs, reporting the material as having excellent qualities but acknowledging the impracticality of producing it at commercial scale, a verdict that every subsequent commercial spider silk venture has ultimately been compelled to echo. The convergence of biotechnology with materials science in the 21st century may finally render this assessment obsolete. The integumentary microstructures responsible for the spectacular iridescence and structural coloration of certain spider species represent some of the most precisely engineered biological photonic structures known to materials science, achieving color production through physical optics rather than chemical pigmentation in ways that have attracted intense research interest from physicists and engineers working on photonic devices and structural color applications. The cuticle of the peacock spider Moretus speciosus has been found to contain arrays of nanoscale protuberances and layered structures that function as diffraction gratings, periodic structures with feature sizes comparable to the wavelengths of visible light that diffract incoming white light into its spectral components and direct different wavelengths to different viewing angles, producing the iridescent rainbow appearance visible to female peacock spiders evaluating potential mates. The spatial resolution of these structural color elements is remarkable. Nanoscale diffraction gratings spanning areas of tens to hundreds of micrometers create color patterns of intricate detail when viewed through the relatively low-resolution compound eyes of female salticids, suggesting that the visual system of the intended audience has co-evolved with the physical structure of the signal to maximize the perceptual impact of the coloration pattern. Beyond their role in spider biology, these biological photonic structures have inspired novel designs for structural color applications in displays, sensors, and anti-counterfeiting technologies, with the principle of angle-dependent spectral diffraction implemented in nanofabricated structures designed for sensing applications where color coding of angular position provides useful information. The thermal biology of spiders as ectothermic animals in variable thermal environments has been studied as a model system for understanding how small arthropods negotiate the challenge of maintaining metabolic function and behavioral capacity across the wide daily and seasonal temperature ranges that characterize most terrestrial habitats. Unlike the endothermic homeotherms that dominate popular understanding of animal biology, spiders have body temperatures that closely track environmental temperature, creating profound challenges for enzyme kinetics and nervous system function at thermal extremes while simultaneously providing opportunities for energy conservation during cold periods through metabolic depression rather than the metabolically expensive maintenance of elevated body temperature. The thermal sensitivity of spider behavioral performance, measured through sprint speed, prey capture success rates, silk construction speed, and courtship display quality, varies considerably between species in ways that reflect both their evolutionary history in different thermal environments and ongoing selection from current thermal challenges. Desert spiders have evolved enzyme systems with higher optimal temperatures and broader thermal performance curves compared to temperate species, allowing functional activity across the extreme temperature ranges of arid environments. Alpine and polar species conversely maintain enzymatic function at temperatures near freezing through structural modifications of enzyme active sites that reduce conformational rigidity without compromising catalytic specificity. Molecular adaptations documented in spider heat shock proteins and metabolic enzymes that parallel similar adaptations in other cold-adapted ectotherms and provide model systems for the engineering of cold-active industrial enzymes. The discovery of ancient spider silk preserved in amber deposits has provided an extraordinary window into the ecology, behavior, and evolutionary history of spiders during the Mesozoic era, revealing webs, silk-trapped prey, and even the behavioral interactions of spiders with their environment at moments frozen in geological time with a fidelity impossible to achieve through any other form of fossil preservation. Burmese amber from the mid-Cretaceous period, approximately 100 million years old, has yielded preserved spider webs of remarkable structural integrity, including spiraling capture threads with regularly spaced droplets preserved in perfect three-dimensional geometry, demonstrating that the modern orb-web architecture with its specialized capture spiral was fully elaborated during the age of dinosaurs. The extraordinary Cretaceous amber specimen of a spider attacking a parasitoid wasp caught in its web, published in 2012 and providing the oldest direct evidence of spider predatory behavior, represents a behavioral interaction frozen at the precise moment of prey capture, confirming that the web-waiting predatory strategy fundamental to modern web-building spiders was fully established by the Cretaceous and has operated essentially unchanged through the subsequent evolutionary changes that have produced the modern spider fauna. Smaller amber fragments containing silk draglines, egg sac wrappings, and substrate attachments have revealed additional details of Cretaceous spider silk biology, including the preservation of attachment disc architecture and dragline thread diameters that closely resemble those of modern spider silks, suggesting conservation of silk biochemistry and spinning apparatus geometry across 100 million years of evolutionary history. The whip spiders, order Amblypygae, while not true spiders, deserve mention within the broader context of arachnid diversity as the creatures most likely to confuse a non-specialist observer into believing they have encountered an extraordinary variant of the spider body plan, and their evolutionary relationship to spiders illuminates the broader diversity of forms available within the ancestral arachnid body plan. Amblypygids possess the characteristic arachnid body organization of prosoma and opisthosoma but have evolved an extremely flattened body form adapted for life in the narrow crevice spaces beneath bark, stones, and leaf litter in tropical and subtropical environments, with their first pair of legs modified into extraordinarily elongated sensory whips that may reach 10 times the body length and serve as organs of tactile and chemical sensation rather than locomotion. Their extraordinary capacity for spatial memory and navigational precision in returning to home crevices from foraging excursions over distances of many meters has made Amblypygae a productive model system for studying navigation in arthropods, with studies documenting path integration, the continuous integration of self-motion information to maintain awareness of current position relative to home, with a precision comparable to the most capable navigating insects. While lacking the silk glands and spinnerets that define true spiders, amblypygids possess chelicerae and pedipalps structurally homologous to those of spiders and venomous scorpions, providing comparative anatomical evidence for the ancestral arachnid body plan from which all these diverse lineages radiated. The silk of the tube-dwelling spiders of the family Atypidae, the purseweb spiders, provides one of the most unusual applications of spider silk technology in the entire order, serving simultaneously as a camouflaged above-ground prey capture device, a sealed protective tunnel providing shelter from predators and climatic extremes, and a structural framework connecting the terrestrial surface to the underground burrow in which the spider spends the majority of its life. The purse web itself is a closed, sealed tube of white silk extending from the underground burrow up through soil or leaf litter and lying flat against the surface of tree bark or rock, appearing externally like a fragment of lichen or bark and providing no obvious indication of its predatory function to passing insects that walk across it. When prey steps onto the silken tube surface, the spider waiting inside detects the disturbance through substrate-borne vibrations, rushes to the appropriate location within the tube, and uses its powerful chelicerae to stab through the silk from below, seizing the prey through the wall of the tube, then cutting open the silk to retrieve the immobilized prey before silk repair specialists within the spider's behavioral repertoire reseal the tube for subsequent use. The silk of atypid purse webs has been measured as particularly resistant to mechanical damage and weathering, properties consistent with its requirement to function for the multi-year lifespan of a single spider without replacement, placing physical demands on silk durability that differ fundamentally from those applied to the routinely replaced webs of orb-weavers. The evolutionary arms race between spiders and their primary insect prey, particularly the diptera, true flies, and hymenoptera, bees, wasps, ants, that constitute the dominant prey of many web-building species, has produced a remarkable array of prey-side counter-adaptations that researchers have only recently begun to systematically document and analyze. Flying insects avoiding spider webs through visual detection represent a well-established selective pressure that has driven the evolution of web decorations, dense bands of white silk added to some webs, whose function has been debated vigorously with proposed roles including bee attraction, bird deflection, and web visibility reduction through camouflage with surrounding bright spots of sunlight, each hypothesis supported by some experimental evidence in different ecological contexts. The cuticular chemical composition of spider webs appears to repel many ant species, social insects that regularly investigate potential food sources through chemical exploration, through a combination of specific deterrent compounds in web silk that have convergently evolved in multiple web-building lineages as a defense against web degradation by foraging ants. Some fly species have evolved silk avoidance behaviors based on detecting the characteristic UV reflectance properties of spider silk, visible to the compound eye UV receptors of many diptera at distances sufficient for collision avoidance, a behavioral adaptation that has in turn apparently driven selection in some spider lineages for web architectures that minimize UV reflectance through alterations in silk protein expression or web geometry that reduces specular reflection toward the blue UV portion of the spectrum. The digestive physiology of spiders represents a fascinating departure from the internal digestion characteristic of vertebrates, with spiders relying primarily on extraoral digestion, the injection or application of enzyme-containing secretions from the gut that liquefy prey tissues outside the body before ingestion as a fluid through the sucking stomach. This strategy, shared with many other predatory arthropods but elaborated to remarkable sophistication in spiders, allows the spider to exploit prey whose external exoskeleton would be mechanically difficult or impossible to swallow whole, by dissolving the internal tissues through the cheliceral puncture wounds while the exoskeleton remains intact as a containing vessel from which the resulting tissue soup is drawn. The digestive fluid injected into prey by web-building spiders contains a complex mixture of proteases, lipases, nucleases, and other hydrolytic enzymes that collectively dissolve essentially all biological macromolecules over periods of minutes to hours, depending on prey size and digestion thoroughness. The sucking stomach, a muscular pump in the prosoma that creates the negative pressure driving fluid ingestion through the narrow pharyngeal passage, operates against considerable hydraulic resistance and has evolved correspondingly powerful musculature in species that feed on large prey whose tissues must be drawn through small cheliceral punctures. Spider feeding efficiency varies considerably with prey size, prey cuticle thickness, and cheliceral architecture, with studies of energy assimilation revealing that spiders typically extract between 80 and 95% of the caloric value of their prey, a digestive efficiency that exceeds that of most vertebrate predators. The spider's silk research conducted aboard the International Space Station and in microgravity experimental platforms has yielded striking and counterintuitive results about the role of gravity in spider web architecture, revealing that the characteristic asymmetry of terrestrial orb webs, with the hub located closer to the top of the web and the area below the hub slightly more extensive than above, is a direct consequence of the spider orienting its body downward while resting at the hub, which in turn reflects the gravitational experience of spiders throughout their evolutionary history. In microgravity, without a clear up-down orientation, spiders initially produced more nearly symmetrical webs with the hub near the center and approximately equal thread density above and below, demonstrating that the architectural asymmetry of terrestrial webs is not a fixed genetic program but a plastic response to gravitational information about body orientation during web construction. The microgravity experiments also revealed that spiders use light as an orientational reference in the absence of gravitational information, tending to orient their bodies toward the light source and producing webs with hub positions displaced toward the light, providing the first experimental evidence that light orientation serves as a backup orientation reference for gravitationally directed web construction under normal terrestrial conditions. These space biology findings have generated productive insights into the sensory integration mechanisms underlying web construction behavior and the evolutionary history of web asymmetry as an adaptation for efficient hub positioning and prey access in normal gravitational environments. The spider community of a single hectare of tropical rainforest in the Indo-Pacific region may contain several hundred spider species representing dozens of families, a biodiversity so extraordinary in its density and functional diversity that the ecological interactions within this community, intraguild predation, kleptoparasitism, substrate partitioning, temporal activity separation, and shared prey competition, constitute a nearly intractable complexity that challenges both observational, documentation, and theoretical modeling. Vertical stratification within the forest is a primary axis of community organization, with species composition changing dramatically between the forest floor leaf litter, the understory herb layer, the intermediate shrub layer, and the forest canopy, each microhabitat supporting specialized communities distinguished by body size, hunting strategy, silk architecture, and activity period. The leaf litter spider community of lowland tropical forests is dominated numerically by linyphiid and oonopid spiders of remarkable miniaturization, some species with body lengths below 0.5 millimeters, that exploit the complex three-dimensional structure of decomposing leaf litter as both hunting territory and shelter, with hundreds of individuals potentially occupying a single square meter of undisturbed litter surface. The taxonomic complexity of tropical spider communities is such that intensive survey efforts in hotspots like the Yasuni National Park in Ecuador have collected specimens belonging to hundreds of spider species in single sampling events, numbers that would take years to formally describe and name with available taxonomic resources, suggesting that a large fraction of global spider diversity remains formally undescribed and that the known catalog of approximately 48,000 species substantially underestimates actual spider diversity. The remarkable chemical ecology of the bolus spider extends beyond its pheromone mimicry of moth species to encompass a secondary luring strategy that further illuminates the extraordinary sophistication of its predatory system. In addition to producing pheromone analogs, Mastophora hutchinsoni has been documented producing compounds that mimic the chemical signals produced by male moths responding to female pheromones, a secondary level of mimicry that attracts male moths already in flight and oriented toward a pheromone source, potentially capturing moths at a different behavioral stage than those attracted by the primary pheromone mimic. The biosynthetic capacity required to produce multiple chemically distinct moth-sex pheromone analogs, compounds that are typically highly species-specific in their precise carbon chain length, functional group positions, and geometric isomer configuration, represents a remarkable evolutionary achievement, as the biosynthetic pathways required for insect pheromone synthesis are fundamentally different from most other lipid biosynthesis pathways and appear to have been co-opted in spider evolution specifically for the purpose of predatory deception. Analytical chemical studies of Mastophora venom globule composition using gas chromatography and mass spectrometry have confirmed the presence of the same or closely related compound classes as the pheromones of target moth species, and have further documented that the specific pheromone mixtures produced by individual spiders match the pheromone blends of the specific moth species most abundant in the spider's local habitat, suggesting a capacity for individual chemical phenotype adjustment. That would require either genetic flexibility in biosynthetic gene expression or dietary acquisition of pheromone precursors from moth prey. The global history of spider taxonomy, the scientific naming, classification, and description of spider species, constitutes one of the most extensive ongoing scientific projects in descriptive biology, with the first formal descriptions of spider species appearing in Linnean taxonomy in the 18th century and the catalogue of described species growing continuously since then at a rate that has accelerated rather than decelerated with advances in technology and the expansion of the global taxonomic community. The World Spider Catalog, maintained by the Natural History Museum of Bern and publicly accessible as a continuously updated online database, currently lists over 48,000 described species in over 4,200 genera and 132 families, numbers that grow by several hundred new species descriptions annually despite representing only a fraction of estimated total diversity. The bottleneck limiting spider taxonomic progress is not primarily the rarity of undescribed species. The majority of any tropical field collection includes undescribed species, but rather the shortage of professional taxonomists with the specialized expertise to recognize and formally describe new species against the backdrop of described relatives, a situation that has been characterized as taxonomic impediment to progress in understanding biodiversity that affects spiders particularly severely given the high diversity and morphological complexity of the order. Integrative taxonomy, the combination of morphological examination with DNA barcoding and phylogenomic placement, has both accelerated the description of new species and revealed extensive cryptic diversity within previously recognized species, with DNA analysis of apparently morphologically uniform species regularly revealing that they contain multiple genetically distinct species whose geographic ranges may differ in ways with implications for conservation management. The mechanical principles underlying the locomotion of spiders across different substrate types, including vertical surfaces, inverted surfaces, water surface films, and extreme inclines, have been studied as model systems for the engineering of bio-inspired adhesive robots and the design of locomotory mechanisms for vehicles that must operate across challenging terrain without continuous contact with level ground. Spider adhesion to vertical and inverted surfaces is achieved through a fundamentally different mechanism than the van der Waals CD-based adhesion of geckos, involving instead tarsal scopulae, densely packed arrays of specialized CD with flat or spatulate tips that achieve adhesion through a combination of van der Waals forces and capillary adhesion mediated by a thin fluid film secreted from tarsal glands. This wet adhesion system in spiders produces forces estimated at many times the spider's body weight per leg in ideal conditions, explaining the seemingly magical capacity of large tarantulas to maintain contact with smooth vertical surfaces, and has inspired the design of engineered adhesive systems using fine fiber arrays loaded with controlled amounts of adhesive fluid. The walking gait of spiders, typically an alternating tetrapod gait in which 4 legs contact the substrate simultaneously in 2 diagonal groups, has been studied using high-speed videography combined with force platforms to measure the ground reaction forces generated by each leg individually, revealing asymmetries in propulsive and braking force distribution that reflect both the spider's center of mass position and the different mechanical properties of the front and rear leg pairs. The representation of spiders in contemporary scientific visual communication through wildlife photography, documentary filmmaking, and microscopic imaging has undergone a transformation over the past decade that has fundamentally altered public perception of spiders in ways that scientific outreach efforts of previous generations failed to achieve. The development of affordable high-quality macro photography equipment, combined with social media platforms allowing direct distribution of imagery to mass audiences without institutional gatekeeping, enabled a generation of amateur naturalist photographers to bring the microscopic world of spider biology to audiences of millions, often achieving images of artistic quality and scientific accuracy that surpassed what professional natural history institutions had previously produced. The work of Jürgen Otto documenting Australian peacock spiders, whose high-definition videos of Moretus courtship displays achieved viral distribution reaching tens of millions of viewers worldwide, serves as perhaps the most consequential example of how citizen science and social media have accelerated public engagement with spider biology, demonstrably shifting attitudes towards spiders in survey populations who reported positive feelings towards spiders after exposure to peacock spider imagery. The scientific community has responded to this democratization of arachnid imagery by engaging more actively with public communication platforms, with researchers including spider biologists directly contributing high-quality educational content to social media channels and achieving levels of public engagement with fundamental arachnological research about silk properties, venom pharmacology, and courtship behavior that traditional scientific publication channels could not approach. The cribellum, the silk-producing organ ancestral to the spinneret-based silk systems of derived spider lineages, provides a fascinating example of how a biological structure can be lost repeatedly in independent evolutionary events while the capacity for its re-evolution apparently persists in the genome for tens of millions of years. The cribellum is a flattened, plate-like organ bearing thousands of minute spigots that collectively produce the woolly, nanoscale fiber hackled band silk used in the prey capture systems of cribellate spider lineages, and it represents the ancestral state of silk production for the order. The sister group to all other spiders, Hypochilidae, retains a functional cribellum, and cribellate species are distributed across many spider families, suggesting that the cribellum was present in the common ancestor of all spiders. Yet many spider lineages, including most of the largest and most species-rich families, have completely lost the cribellum over evolutionary time and replaced hackled band silk capture systems with the chemically adhesive wet silk systems characteristic of ecribellate spiders. This repeated loss appears to reflect genuine adaptive advantages of wet adhesive silk over dry cribellar silk in certain ecological contexts. Wet glue maintains adhesion in damp conditions better than cribellar silk, and the energetic cost of producing cribellar silk through the elaborate combing process using the Calamistrum seedi may be higher than producing equivalent function wet glue in some environments. The genomic mechanisms preserving or eliminating the capacity for cribellum expression represent an active area of evolutionary genomics research with broader implications for understanding how complex phenotypic innovations are gained and lost over evolutionary timescales. The spider fauna of caves, the troglobitic and troglophilic arachnid communities that inhabit the perpetual darkness of subterranean environments, represents one of the most biologically distinctive and least studied components of global spider biodiversity, with dozens of highly specialized cave-adapted species described from cave systems worldwide and an unknown but certainly very large number remaining formally undescribed. Cave-adapted spiders exhibit the classic suite of troglomorphic modifications seen across taxonomically unrelated cave-adapted animals: progressive reduction and eventual loss of eyes and optic ganglia, loss of pigmentation producing a ghostly white or yellowish body color from the absence of melanin synthesis, elongation of sensory appendages particularly the tactile seedi and first pair of legs, slowed metabolism and extended lifespan relative to surface relatives, and behavioral modifications including reduced activity budgets, extended developmental periods, and reduced reproductive rates reflecting the low and unpredictable prey availability of cave ecosystems. The dramatic convergence of troglomorphic characters across independently cave-colonizing spider lineages from taxonomically disparate families provides one of the most compelling examples of parallel evolution available within the order, demonstrating that the same suite of morphological and physiological modifications represent optimal adaptations to cave conditions regardless of which spider family happened to colonize a particular cave system. Some cave-adapted spiders of the family Tetrablemidae in Southeast Asian cave systems have been described with apparent complete loss of the visual system, including not just the eye lenses but the entire optic lobe regions of the brain, representing a dramatic anatomical reduction that illustrates the power of relaxed selection on structures costly to maintain in the absence of the visual information they normally process. The relationship between spider venoms and the pharmacological treatment of chronic pain represents one of the most productively explored intersections of natural product chemistry and clinical medicine in contemporary pharmaceutical research, with several spider venom peptides either in clinical development or serving as molecular templates for the design of synthetic analogs with improved pharmacokinetic profiles. The particular value of spider toxins as analgesic leads derives from their evolutionary specialization in targeting the specific ion channels and receptors most implicated in pain signaling: voltage-gated sodium channels NaV 1.7 and NaV 1.8, voltage-gated calcium channels, and acid-sensing ion channels, with a selectivity and potency developed through hundreds of millions of years of optimization against the ion channel profiles of real prey and predator nervous systems. The tarantula toxin ProTX2 from the spider Thrixopelma pruriens selectively inhibits NaV 1.7, the sodium channel subtype most strongly implicated in inherited pain insensitivity syndromes in humans and consequently the highest priority drug target for pain management, with remarkable selectivity over other sodium channel subtypes, making it a valuable template for analgesic drug design. Australian funnel web spider venoms have yielded Hi1A, a peptide inhibitor of acid-sensing ion channels, which has demonstrated extraordinary neuroprotective properties in animal models of stroke, providing a completely unexpected therapeutic application for a toxin whose natural function is prey paralysis, and has progressed toward clinical evaluation as a potential stroke treatment in patients administered the peptide within therapeutic time windows following ischemic events. The evolutionary genomics of spider silk genes, the spidroin gene family, has revealed a history of gene duplication, divergence, and domain shuffling that illuminates how molecular evolution generates functional diversity within a gene family through combinatorial variation of a relatively small number of conserved structural elements. The spidroin genes are characterized by extraordinarily long, highly repetitive central encoding regions flanked by much more conserved terminal domains, a molecular architecture that creates unusual challenges for standard sequencing technologies while generating the protein structural features responsible for silk's remarkable mechanical properties. Phylogenomic analysis of spidroin gene diversity across spider lineages has revealed that the major silk types, major ampullate, minor ampullate, tubuliform, flagelliform, aciniform, are each encoded by distinct gene subfamilies that diverged through duplication events occurring at specific points in spider evolutionary history, with the emergence of new silk types corresponding to the evolution of novel predatory or protective behaviors requiring silk of new functional properties. The flagelliform gene, encoding the elastic capture spiral silk, appears to have emerged relatively recently in spider evolutionary history, present only in the ecrobellid orb-weaver lineages that have replaced cribellar's silk capture systems with wet adhesive spirals, suggesting that the evolution of flagelliform silk was a key innovation enabling the extremely successful wet adhesive orb web strategy that characterizes arachnidae, tetragnathidae, and their relatives. Whole genome sequencing of spider species is rapidly expanding the genomic resources available for spidroin evolutionary analysis, with genomes now available for multiple theraphosid, araneoid, and other spider lineages revealing levels of spidroin gene family complexity that continue to surprise researchers. The discovery that many spider species produce ultraviolet reflecting silk, detectable to bees and many other insects whose vision extends into the near ultraviolet, has fundamentally complicated the long-held assumption that spider webs function as passive, invisible traps for flying insects, revealing instead that web visibility to potential prey may be an adaptive rather than maladaptive feature of web construction in some ecological contexts. Research in the 1990s and 2000s demonstrated that the structural decorations added to some orb webs, bands, spirals, and disc-shaped stabilimenta of white silk, sometimes called writing, reflect ultraviolet light strongly, making them highly conspicuous to bees and flies whose vision spans the UV range, and raised the initially counterintuitive hypothesis that these decorations attract insects to webs rather than deterring them. The UV attraction hypothesis for stabilimenta received experimental support from studies showing that webs with stabilimenta caught more UV-sensitive prey in some studies, and from observations that stabilimentum construction rates increase in spiders maintained under prey-poor conditions, consistent with an interpretation of stabilimenta as active prey-attracting devices rather than mere byproducts of silk behavior or camouflage elements. The diversity of stabilimentum forms across different spider species, ranging from simple discs to complex spiral bands to radial crosses in different genera, and the considerable variation in stabilimentum presence and morphology within species depending on habitat, season, and prey availability has generated a rich and not entirely resolved scientific debate about whether stabilimenta serve primarily as prey attractants, as camouflage against complex backgrounds, as bird deflection signals, warning birds of the web's presence, or as byproducts of the spider's own thermoregulatory silk handling behaviors. The ecology of spider egg sac construction and the diversity of strategies employed by different spider lineages for protecting eggs from desiccation, thermal extremes, parasitoids, and egg predators constitutes one of the most functionally diverse domains of spider's silk application, with egg sac architectures ranging from simple wrappings of a few layers of loose silk to elaborate multi-layered structures of exceptional mechanical complexity. The tubuliform silk used specifically for egg sac construction is the most biochemically distinct of all spider silk types, with spidroin sequences characterized by unusually long, variable amino acid motifs in the repetitive central domain region compared to other silk types, and mechanical properties optimized for compression resistance and water barrier function rather than the tensile strength and extensibility that characterize dragline and capture thread silks. Many spider species camouflage their egg sacs through the incorporation of plant fragments, soil particles, or in some cases fecal materials into the outer silk layers, a behavior that requires significant additional behavioral investment in material collection and integration but provides substantial reductions in parasitoid wasp attack rates documented in carefully controlled field studies. The evolution of maternal egg sac guarding, active defense of the egg mass by the female against approaching predators and parasitoids, has arisen multiple times in different spider lineages and represents a significant extension of silk-based protection with behavioral investment, with guarding effectiveness studies demonstrating that attended egg sacs suffer substantially lower parasitoid attack rates than experimentally abandoned sacs in species where both conditions can be created under controlled conditions. The scientific and philosophical legacy of Aristotle's observations on spiders, documented in the Historia Animalium written approximately 350 BC, establishes arachnology as one of the oldest continuing traditions within the broader history of natural history observation, with passages describing spider silk production, web construction, prey capture, and egg sac construction that demonstrate careful direct observation of spider behavior rather than mere compilation of received traditions. Aristotle's description of the spider's web-building process in the Historia Animalium, noting that the thread emerges from the spider's body interior and that webs are constructed sequentially from frame threads to spiral, is sufficiently accurate to be recognizable as orb web construction despite the absence of modern optical instruments or systematic experimental methodology. The zoological tradition initiated by Aristotle was extended through medieval encyclopedists, Renaissance naturalists, and eventually the systematic taxonomy of the 17th and 18th centuries into the formal scientific discipline of arachnology as practiced today. The transition from descriptive natural history to experimental arachnology is typically dated to the work of the Swiss naturalist Johann Caspar Sulzer and the French naturalist René-Antoine Foucault de Riaumur in the 18th century, who performed controlled experiments on spider biology rather than merely observing and recording, and whose experimental approaches to questions about silk production, prey capture, and web function prefigured the experimental arachnology that became standard practice only in the 20th century. The continuity of intellectual engagement with spider biology across 2,400 years of documented scientific inquiry speaks to the persistent fascination exerted by these animals on scientific minds across vastly different cultural and intellectual contexts. The spider Steatota grossa, the false black widow, exemplifies the epidemiological complexity surrounding spider envenomation in human populations, as a species whose medical significance has been substantially overestimated in popular literature, partly because of confusion with the genuinely dangerous Latrodectus widow spiders whose webs and body form it closely resembles. Steatoda species produce a venom containing a family of proteins called steatodotoxins that cause a milder syndrome than true latrodectism, characterized primarily by localized pain, regional swelling, and occasionally nausea, but whose similarity to widow spider bites in symptomatology has led to consistent misattribution of Steatoda bites as Latrodectus bites in clinical and epidemiological records, particularly in regions where Latrodectus is presumed present but is actually rare. The ecological success of Steatoda species in human-modified environments, particularly heated buildings, garages, and storage spaces, means that human encounters with these spiders are genuinely frequent. Yet, epidemiological data suggest that bites are rare despite high cohabitation densities, reflecting the generally non-aggressive nature of Steatoda and their tendency to flee rather than bite when disturbed. The challenge of accurately attributing spider bites to specific species in clinical settings, where the spider is rarely captured and brought to the treating physician, and where the presenting clinician may lack arachnological expertise to identify the responsible species even when a specimen is provided, represents a fundamental impediment to accurate understanding of the medical significance of different spider species, with consequences for both clinical management and public communication about spider hazards. The ecological role of spiders as prey, not merely as predators, in terrestrial food webs constitutes an underappreciated but functionally significant component of their contribution to ecosystem energy flow, with spiders serving as important prey items for birds, lizards, centipedes, parasitoid wasps, and a remarkable variety of other spiders in feeding relationships that connect the invertebrate and vertebrate worlds through trophic linkages of considerable ecological consequence. The importance of spiders as prey for insectivorous birds during the breeding season has been particularly well documented through brood diet studies revealing that nestlings of many passerine species receive spiders as a disproportionate fraction of their diet relative to spider abundance in the environment, a dietary preference apparently reflecting the particularly high protein and taurine content of spider bodies compared to other invertebrates, with taurine being an essential nutrient for normal visual system development in altricial nestling birds. The mutual predation between spider species, intraguild predation between web-building spiders that may invade each other's webs, between cursorial spiders hunting other spiders, and between parasitoid-like specialists such as the pirate spiders of Mimetidae, creates a complex matrix of negative interactions within spider communities that strongly influences community structure, species composition, and the body size distributions of coexisting spider species. The regulation of spider population density through these combined predation pressures from both higher and equivalent trophic levels means that spider populations in most ecosystems are subject to both bottom-up regulation through prey availability and top-down regulation through predation, a dual control structure that generates complex population dynamic behaviors not easily captured by simple prey-dependent or predator-limited population models. The mythological spider traditions of Oceania and the Pacific Islands constitute one of the richest and least widely known bodies of arachnid cultural material in world tradition, with spider figures occupying cosmogonic, creative, and navigational roles across the diverse cultures of Melanesia, Polynesia, and Micronesia that reflect both independent invention and historical connections among island cultures spread across the world's largest ocean. In Melanesian traditions, particularly among cultures of the Solomon Islands and Vanuatu, the spider is often associated with the trickster-creator archetype, a being of original power who shapes the world through cunning manipulation of available materials in ways that parallel the spider's physical manipulation of silk into architecturally sophisticated structures. The navigational spider mythology of certain Polynesian cultures is particularly remarkable in its integration of actual spider biological observation into cosmological narrative. Spiders, observed ballooning on their silk threads and using wind currents for long-distance travel across open ocean surfaces, became mythological models for the human navigation of open sea in traditionally navigated voyaging canoes, with the spider's silk thread conceptualized as the invisible connection between the voyager and the home island that could guide return navigation in the absence of visual landmarks. The Tok Pisin word for spider in Papua New Guinea, where over 800 distinct languages are spoken and spider cultural traditions are correspondingly diverse, has been documented in several communities associated with creative power and wisdom in ways that echo Anansi's creative role in West African tradition without any historical connection, suggesting a genuine convergent cultural attribution of creative intelligence to the spider's observable building behavior. The burgeoning field of spider phylogenomics, the use of genome-wide sequence data to reconstruct the evolutionary relationships among spider lineages with unprecedented resolution and statistical support, has overturned several long-held assumptions about spider evolutionary history while confirming others, producing a tree of spider life whose broad outlines are now becoming clear even as many detailed relationships within families and between closely related families continue to be resolved with increasing data. The placement of the jumping spiders, Salticity, as the sister group to a diverse clade including the cobweb spiders, sheet web spiders, and orb-weavers rather than as an isolated basal lineage reflects a fundamental revision of previous morphological classification and has important implications for understanding the evolution of the sophisticated visual systems and complex courtship behaviors that characterize salticids. The repeated independent evolution of orbwebs, once thought to be a single synapomorphic innovation of a clade called orbiculary, has been revealed by phylogenomic analyses to be more complex than originally recognized, with some analyses supporting the single origin hypothesis while others support multiple origins. A question of particular evolutionary significance because it determines whether the full elaboration of orb-web construction required a single innovation of enormous complexity or whether this complexity arose independently multiple times. The incorporation of transcriptomic data from silk glands and venom glands into phylogenomic analyses has added an additional dimension to spider evolutionary reconstruction, allowing the molecular evolution of silk and venom gene families to be mapped onto the species phylogeny and revealing episodes of accelerated diversification that may correspond to ecological transitions in predatory strategy or web architecture. The conservation biology of spiders, the assessment of threat status, identification of critical habitats, and development of management strategies for spider species at risk remains substantially less developed than equivalent efforts for vertebrate and plant taxa, reflecting both the taxonomic impediment that limits the description of new species and the general public and institutional preference for charismatic megafauna as conservation priorities, yet the ecological significance of spider communities in terrestrial ecosystems provide strong pragmatic arguments for investing in spider conservation alongside other invertebrate taxa. The IUCN Red List currently includes formal threat assessments for only a small fraction of described spider species, a number that represents a tiny proportion of total spider diversity and that is constrained by the fundamental requirement that Red List assessment requires sufficient information about population size, trends, and geographic range to apply the established assessment criteria. The conservation of spider diversity in practice depends primarily on habitat conservation, particularly the preservation of old-growth forest, natural grassland, and cave habitats that support the most specialized and least dispersal-capable spider species, rather than species-specific management interventions, as the small body size and high species richness of spiders makes targeted recovery programs for individual spider species logistically impractical except in the most extreme cases, such as island endemic species with severely restricted ranges. The Hawaiian happy face spider, Theridion grallator, so named for the smiley face pattern on the opisthossoma of some individuals, has become one of the most extensively studied spider species from a conservation and evolutionary genetics perspective due to its restricted range on threatened Hawaiian forest habitats, its remarkable color polymorphism maintained by natural selection, and its value as a model for understanding the evolutionary dynamics of small island populations subject to habitat fragmentation. To contemplate the spider in full, its 380 million year geological tenure on this planet, its silk more sophisticated than any human engineering, its venom a pharmacopoeia of undreamed molecular specificity, its behaviors spanning from reflex-mediated simplicity to apparent cognitive flexibility, its cultural presence woven through every human civilization that has shared its world, is to confront one of the most humbling realities available to the naturalist's attention, that complexity, sophistication, and ancient wisdom are not the exclusive properties of large, warm-blooded, vertebrate creatures whose faces meet ours with a legibility we find reassuring. The spider has watched the continents drift and reassemble, has survived extinction events that erased the dinosaurs and reset the trajectory of vertebrate evolution, has colonized every terrestrial environment from polar ice to equatorial rainforest to the lightless galleries of subterranean rock. It has inspired some of humanity's oldest stories, some of its most enduring mythological figures, and some of its most productive scientific inquiries. And it continues, in the dew-spangled webs of every autumn morning and the patient waiting of every cellar corner, to be precisely what it has always been, an architect of extraordinary silk, a predator of ancient lineage, a presence in the living world of such deep time and functional perfection that our brief scientific engagement with it is, by any honest measure, still only just beginning. What remains undiscovered in the spider's world dwarfs immeasurably what we have so far learned, and that fact alone is sufficient reason for continued wonder.